Hello, hello, hello. Welcome, welcome. Hello. How are all of you? It has been a while since I have been here behind this screen. I have been upgrading the setup here. No longer will I be streaming on the Phytop. We are now on an actual desktop. Yes, the delay is much better. Yes. Gone are the days of suffering through streaming with a laptop. Now we are firmly in desktop zone, which is fantastic for me. Chat's a little big. Shove you in the corner right there. Yeah, it is such a breath of fresh air to be streaming, especially on a setup that doesn't completely chug and increase the temperature of the room. So thank you to everybody who helped me set up my computer. It was not a trivial task. I had quite a bit of help from community members in my Discord. But yes, I am very, very happy to be streaming in a way that is reasonable. I can't believe I streamed for so long just with that laptop. It's pretty incredible. So welcome to you. Welcome all of you to the first of the Science Trivia Nights. Very exciting. Hello, Mr. Fox. Hello, Neutrophil Mapping. Of course, hello, Surprise Pants and John the Eosinophil. The Wolf. Hello, Dark Cube. Hello, More Juice, Thomas the Thermonuclear Bomb, of course. Welcome all of you. So exciting. Hello, Kevin. Welcome. Today, what we are going to be doing is we are going to be playing a bit of trivia. Now, I have been hard at work at making the pre-on video, which again is also something that's been going very, very well after upgrading to a computer with an external GPU. Let me tell you, not having to wait 10 minutes for Adobe Premiere to do literally anything is just a godsend. It's been pretty incredible. So I hope to get the pre-on video out by the middle of July. So I've got, I think, maybe 8 more minutes of video to finish, and then by then I'll finally be able to release this dang pre-on video. I'll finally be able to do like 1,000 journal clubs on pre-ons, because the science behind pre-ons is really cool. I'm very excited. Hello, Engine Crafter. Welcome to the stream. Just saying how much I appreciated the help from the Discord, which, by the way, as of right now, the Discord is still kind of private. If you join membership, you can have a link to join the Discord, as I am still slowly working my way up to being confident that the thing is not going to be instantly destroyed by bots. But, you know, we'll see. All right, let's just get right into it. We're going to play a little bit of trivia, and the way that this trivia is going to work is I'm going to ask you all 6 biology questions, and I have picked 6 questions from the chat. If your questions don't appear here, it has the highest priority in the next stream, if I deem that they are appropriate for something like this. So we can engage in a friendly battle of the brains, 6 questions for you, 6 questions for me. And of course, the questions that I pick are basically excuses for me to talk about cool biology stuff. Of course, you can participate in those bonus questions, but, you know, they won't count for our competitive score. What if the bots figure out membership? We're all doomed. If bots want to give me money, you know, I'm not too mad about that. Let's go ahead and put on some Lo-Fi Girl. Let me know if the music volume needs some adjusting. Otherwise, let's just get right into it. I'm very excited to play some trivia. All right. So let's go ahead and get this started. Let's see if I can run a poll in OBS. Doesn't look like I can. No. Okay, that's fine. Okay, so the first question we have here. Get my timer out. Yeah, your boy's got a timer up. The first question, of course, I'm going to start with easy questions. This is an easy question. Which of the organelles contains destructive digestive enzymes? So let me go ahead and set up that poll. A, B, C, D. All right, go ahead, and I'm going to start the timer. Yeah, we've come so far. That's right. We do have a timer. Make sure to answer via the poll and not in the chat. That's an accelerated GPU timer. True. We're going places. It's also because of the support that I've gotten on this channel is really incredible, because I'm someone who is very hesitant to invest in my hobbies, like money-wise. So I made the plunge to actually upgrading my setup because I started to see, hey, you know, maybe this hobby can sustain itself instead of me just dumping a lot of money into it. So thank you all seriously so much for your membership and for just looking at my videos, especially if you have ad block off, contributing to helping me build up the quality of the content that I'm able to produce, which is very exciting. Quality is so much better. I know. Thank you so much. All right, that is the timer. Let's go ahead and close out that poll. How did you all do? So we'll go ahead and we'll end the poll. 67% picked the most common answer, which was D. Chat, you are off to a good start, although there is a bit more heterogeneity. It is, in fact, D, the lysosome. Very good, very good. I wish I could join membership, but I can't. Don't worries. No worries. If you watch my stuff, you are contributing to the funds that I use to make the channel better, which is something that I appreciate. So, what is the lysosome? So the lysosome is a little pocket of horrible, nasty things, including a lot of acid and enzymes that break down things like proteins and lipids. And so what I have here is an image of cells. I think these are HeLa cells. So if you know about HeLa cells, that's what these are. HeLa cells, so these are immortalized human cells. And so these have been stained, labeled fluorescently with different markers. In blue, we have DAPI. This is the most common stain for the nucleus. So this is where all the DNA lives, right here in blue. In green, I believe we have a stain for, I think it's actin in green, but I'm not entirely sure. It's some component of the cytoskeleton, so that gives you an idea of the shape of each one of these cells. And in red here, we have the lysosomes. So they are stained with an antibody against a lysosomal compartment, and then an antibody that fluoresces red was attached to those antibodies. And so each one of these little dots, we call them punctates if you want to be fancy, represents one lysosome, so one packet of these horrible digestive enzymes. And so if you've seen a couple of my videos, you know that lysosomes often have a role, especially for professional phagocytes like me, neutrophils, and macrophages, to act as sort of the cell's digestive system. So if you see an immune system cell kind of trolling the body looking for bacteria to eat, they will likely be digested by fusing with this organelle called the lysosome, which is very, very interesting. Welcome, Clockwork. It's a trap chat. It's actually a series of lectures, hidden side trivia questions. I don't disagree. Yeah. Not surprised that decades down the line, someone will try to clone Henrietta when it becomes possible. I'm not entirely sure how well these HeLa cells have maintained Henrietta's original genome, to be honest, because they've been passaged so many times. Are lysosomes like cellular self-destruct end systems? That's a good question. I'm not entirely sure how to detail the link between lysosomes and apoptosis, sorry, pronouncing that correctly now. I typically only think of them in the context of digesting things like germs or proteins and stuff like that. Phy loves eating germs. True, true. I'm definitely acing science with this one. I always say that my channel is not a substitute for a proper biology education, and I know that most of my audience gets that, but really, you should go to school. You should attend your classes. HeLa cells are technically not human anymore. Interesting. I've not heard that particular point. It'd be really cool if you could elaborate on what you mean by that. Their genome drifted too far. Interesting. Cool, cool. Apoptosis is governed by mitochondrial control pathways and a few other things. The thing is, apoptosis is so complicated that I only typically think about the initiation of apoptosis. What happens afterwards, I actually don't remember, which is very cool. Engine Crafter, remember for 4 months. I can't believe it's been that long. Thank you for continuing to support the channel. It's awesome. They're more of a toolbox for things for demolishing cells. Exactly, exactly. Very cool things. Wish I knew a little bit more about them, but I just wanted to give these little things a shout out, and also to show off some fluorescence microscopy, because this kind of image is, I think, some of the cooler stuff that you can find in biology. Very neat. All right, good job, chat. Let's go with the next question. Which of the following terms refers to a variation of a gene? So this is another easy level question. If you've ever taken a biology class, you should be able to get this. And so we'll go ahead and we'll start that poll. A, B, C, and D. Go ahead. And I'll try to reset this timer, although this particular one... Oh, timer! Timer, why? No, the timer's gone! I got to use the timer one time, and it's popped out of existence. That's no good. I mean, you can have all the time that you need, I suppose, chat. I truly don't understand. I see the timer on my end, but it doesn't appear to be showing on yours, which is pretty concerning and something that I'll probably have to work out later in the future. Finally, no buffering, the new fire station slaps. Yes, yes. The timer was too much for your GPU, maybe. Looking at prebiotic yogurt, try not to eat germs. Yeah, yeah. See my kombucha video for my thoughts about probiotics. Oh, I know, it's finally no buffering. Such a good feeling. I'm so happy. And again, it's thanks to all of you that I'm able to stream in this quality. But yeah, the scuff is back. I don't know what happened to the timer. I tested it before stream, I promise. Okay, that is all the time that we have for that poll. Let's go ahead and end that poll and see how chat did. All right. Looks like we have a majority answering B. Let's go ahead and reveal the answer. B, allele. Yep. Genetic type is just something that I made up. Nucleotype is something that I'm pretty sure I also made up. Genoform is something I completely made up as well. Phytop scuff has been eliminated, so now there's more room for human phi scuff. You mean neutrophil phi scuff, of course. All right, so I want to talk about alleles. So alleles are different variations of a gene. And so you typically, a gene is made of DNA, and it's a very specific DNA sequence which codes for a specific protein. If you change that sequence of DNA, you change the protein, right? And so different forms or different variations on that genetic sequence can result in slightly different proteins. But when we talk about phenotypes are visible things. So things that produce visible effects in an organism. So if you have a couple of different variations that might not result in anything different about your lived being, then that might not be counted as something that is visibly phenotypic. I want to talk about a very polymorphic gene, one that contains many, many alleles, and that is the protein called MHC. It's actually a protein that I have worked on. I've done a bit of research into this. It's a protein that is very near and dear to me. And so I want to get a little chance to talk about this. So MHC, it's something that I talked a little bit about in my videos. It's a protein that acts as a signaler to the immune system to inform the immune system whether or not a particular cell has been infected or is healthy. It can display self and non-self antigens. So self antigens are pieces of proteins that come from proteins that you naturally make. And if a T cell, as shown in this slide, if a T cell binds to a self antigen, then the T cell is chill, doesn't do anything. It says, okay, we have a normal cell here. But if this protein displays a non-self antigen, say from a virus or from a bacteria, then the T cell can recognize that it has bound a non-self antigen and then go off to do T cell things. So this protein, MHC stands for Major Histocompatibility Complex. And then this particular one is the first family, so MHC1. So the way that this protein does this is that it has this little peptide groove. So on the left here, we have this cartoon version. You see the subunits, the pieces of the protein. And then on the right here, we have the cartoon version. So you have ribbons for alpha helices and these arrows for beta sheets. And then these spaghettis are regions of the protein that are flexible. And if we go, so this is like the same view, right? But if we rotate it towards the audience and look at it from the top up here, actually, I don't know if you can see my mouse when that's unfortunate. Give me 1 second. See if I can actually show my mouse so that I can actually point to things. Yeah, it says capture cursor, but I'm not really seeing my cursor. That's a little unfortunate. On the right over here, this is the top view of that protein, of the major histocompatibility complex 1 protein. And you see this orange spaghetti, this orange noodle in here? This orange noodle is an antigen. So this is a piece of a protein that was digested by the cell that can be displayed in what is called a peptide groove. So, I'm reading back chat for a sec. Related to histamines and stuff. Well, I think they both refer to the study of immunity and antibodies and stuff like that. So I think that's the common root, but I'm not entirely sure. Thank you, Clockwork, for helping out. Yeah. So you see in the peptide groove, you have this little orange spaghetti. But the thing is that every single one of you has a unique footprint of immunity, right? Each one of our immune systems is slightly different. This is by design. This is by design because you can imagine that if all of our immune systems were exactly identical, then one potential plague could wipe all of us out. And that would be very bad. It's actually a very good thing that all of our immune systems are slightly different. And the way that they're slightly different, part of the way that they're slightly different is through this particular gene. MHC, Major Histocompatibility Complex 1, is a very polymorphic gene. All of our MHCs, the combination and the variations that we have are all slightly different. And where they're different is in this peptide binding region, this groove where you can bind this orange little spaghetti. Each one of our grooves is a little different, so the antigens that our MHC can display, they're all just slightly different. And this helps us hedge against things that could eliminate us, like plagues and stuff like that, which I think is very neat. So I think that it's a cool chance to talk about something that is multi-allelic, this wonderful protein keeping us all alive and on our toes for potential infection. Okay, chat, you are on a roll. So far, you are hitting it out of the park. Now we're getting into things that are a little more esoteric. So those two, if you've ever taken a biology class, you could get them right. I shouldn't say should, but you could. Now, these are questions that are a little harder. They're a little harder, which might require a bit more extra schooling or a bit more intense recall, we'll say. So I'll go ahead and I'll start the timer on my end because you can't see it on your end for some reason, which is very sad. But go ahead and give that a shot. I can't believe this timer only worked one time. That's pretty bonkers. No Googling, chat. Yeah, exactly. What's the fun in that? the thing is these are basically clicker questions, but for fun, right? And oh, I want to do a meta teaching stream at the start of the school year. So sometime in August or September, I want to do, because I am a professional teacher. Sounds weird to say it that way, but I do teach and it does earn me an income. It'd be really interesting, I think, to talk about the hidden secrets behind the research on how students learn, which I think is going to be quite relevant for students starting their semesters. I wonder if that sounds interesting to you. And for those of you who are career professionals right now, I think it's still interesting to learn because, I don't know, you might be able to like, I don't biohack your own brain, or we should probably not call it that. Alrighty. Okay, that's all the time you've had, chat. Let's go ahead and see how you did. This is a question about metabolism, specifically the citric acid cycle, and we've got expectedly a bit more heterogeneity here. So we have 62% saying pyruvate. Now that is very, very interesting because I think that indicates that the majority of you have had some level of deeper biology education because this is definitely not something that people would normally remember if they chose not to go into biology. So that's very, very fun. Biohacking is a trigger word for this channel file. Yeah, I know. I slipped up. Yeah, it looks like that a good number of you have picked my bait answer, the citric acid citrate. That's technically not the input for the citric acid cycle. It's technically the first step. So I'll go ahead and show this pathway. And then I'm going to get into something that is perhaps more interesting. It's my commentary on the education aspect of this cycle. The word pyruvate just jogged my high school brain PTSD. Maybe that's it. And I'm about to talk about PTSD referring specifically to having to learn this in biology. I knew it was too obvious, but I picked it anyway, like a sucker. I mean, you know, sometimes it happens to the best of us. So this is the citric acid cycle. It is an intermediate between glycolysis, which is the breakdown of sugar, to oxidative phosphorylation, cellular respiration, which happens in the mitochondria. And before that can happen in the mitochondria, you need to first extract some protons and some energy intermediates so that you can actually use the ATP synthase pump that Clockwork here in the chat has lovingly dedicated a whole video to. You should definitely check out Clockwork. Clockwork, if you're still here, if you could plug your channel, that would be awesome. And so if you see up here, the input to this cycle is actually pyruvate, or acetyl-CoA is also an acceptable answer, but I figured pyruvate would maybe be a little easier for people to remember. Citrate's actually the first step, so it's a little misleading, but there you go. Citric acid was the only compound I recognized. I mean, you're not alone. This is something that is like deep biology lore, I guess you could say. Where does this cycle happen? I believe the citric acid cycle happens in the cytoplasm, but I'm not entirely sure. That is my guess. I don't remember everything perfectly about this too, and I'm actually going to talk about why that's okay. I have this jar full of citric acid powder that I use for cooking. Yeah, that kind of stuff can be used to flavor sour candies, just very exciting. I actually don't know how to plug links on chat, y'all. Well, click on Clockwork's channel and you'll see some very high quality biology animations. Very, very high quality stuff. And Clockwork and I actually have a collaboration that we are planning to talk specifically about protein. So keep an eye out for that. What does the mitochondria do then? The mitochondria has enzymes that specifically are in the actual body of the mitochondria that can use the protons generated from the citric acid cycle to drive a pump that grinds ATP together is the best way that I can put it. Okay, so here's my rant space. Okay, so people on my Discord are familiar with this little rant that I had. There was a post on our biology about how seemingly reductive and stupid it is to have every biology student learn the citric acid cycle, glycolysis, cellular respiration, etc., right? And I understand. This is a person who made this post was interested in the natural sciences. So natural sciences meaning studying things like animals and plants and insects and stuff like that, and ecology. And they were lamenting the fact that they had to go through all this biochemistry, which seemed very difficult. And part of me is very sympathetic to that idea, because even I, as like a professional biology person, I don't know everything about glycolysis or the citric acid cycle or how the mitochondria makes ATP. I know enough to get by. And the thing about having a robust education in the molecular sciences is that it leaves you with fewer possibilities for making incorrect assumptions, right? So I know enough to be dangerous. Well, we did talk about cyanide, I think, last time we talked about the mitochondria, so, you know, watch out. Mitochondria is an incredibly complex organelle, so much more than a powerhouse. Yeah, we could talk about mitochondria for eons, and also mitochondria play a very interesting role in the propagation of infection, right? A lot of germs in your body love to get a hold of that mitochondria and do all sorts of nasty things to that mitochondria to give themselves more energy instead of you, which is really, really interesting, right? And so I hear this a lot, this sentiment that we should not be focusing this much on brute force memorization of all these complex pathways, especially from biology students, some of which I have taught before, because it seems like you don't really need to know this for the most part. Like, do you need to know the citric acid cycle unless you're studying something like metabolism? Yeah, probably not. But the thing is for all of you who are entering high school or in undergrad or entering undergrad, the utility of college is not necessarily learning facts, right? The utility of college is a safe place for you to teach yourself how to learn. And I think that doesn't really get taught enough. You learn facts for sure, and those facts are important, but the thing that your money is actually worth is the ability to teach yourself. Because honestly, if you want to just know about biology, you can just buy like Khan Academy, not a sponsor, right? You could just watch videos and learn things about biology. But if you want to be a biologist, you have to know more than just facts. You have to learn how to think like a scientist. And so forcing yourself to learn through all of this is a way for you to train your brain to handle a form of information that it might not have been comfortable with before. And believe me, I am sympathetic because organic chemistry crushed me when I was an undergraduate, but I am thankful because the more you force yourself to learn things that are hard, the smarter you become, even if you never use that knowledge in the future. Yeah. All right. So that's my rant space. If you're a biology student, learning the citric acid cycle is just like brushing your teeth. You gotta do it. You might not love to do it, but you gotta do it. It's good for your brain. All right, question 4, chat. Which of the following is not a germ layer during vertebrate embryonic development? Let's talk a little bit about development. A field of biology that I have often plugged, but I am too scared to cover live. I love developmental biology, but I'm also hot garbage at it. I think developmental biology for me is a particularly bad match because my memory is so bad. My memory, my short-term memory, I can only fit 5 things in my short-term memory. So I don't know if you've ever done a short-term memory test, but I've talked about this enough on stream before. My short-term memory is very, very small. Cannot remember phone numbers to save my life. And so developmental biology, where a lot of it is memorization, was really tough, but I remember learning about it in graduate school. It was my worst class, but it was so incredible, and so this is a really exciting excuse for me to be able to talk about developmental biology. Yeah, this is difficult. Yeah, you would only probably know this if you've taken a biology class recently. This one's also easy if you were Catholic enough to be first to learn Greek and Latin. That's kind of interesting because of how I chose some of these answers. But you also have to take math and chemistry for biology to get easier. Yeah, so unfortunately for fans of classical sciences, every field tends towards quantification. And so, unfortunately, you were born in the current time period and you need to know how to do math. That's just the reality of science nowadays. But how is this medium? Well, I actually don't remember. I think this is covered in AP Biology. I think. It's been a while since I cracked open my AP Bio textbook, but I think this is covered in AP Biology. Because this is covered in Intro Bio, so it should be also in AP Bio. Okay, let's see what chat said. This one was close. This one was close. 56 choosing C, exoderm. I think we impressed Phi too hard in the previous stream, and now he thinks chat is full of geniuses. I talk about that all the time with Toxo, actually. Both of us remark how smart my chat is. I was actually so blown away by the fact that you all got... You outdid me in trivia last time we played. So maybe I did scale the difficulty a bit, but chat, you did still get the correct answer. It is the exoderm. Kind of a mean question when everything ends in derm, and so I just had to throw another suffix there. But well done, chat. Even though this is harder, you are all still crushing it as a collective right now. So good job to you all. So what are these germ layers? So developmental biology, a lot of it is about how you go from sperm and egg to a whole organism, and that process is so complicated, but it's so beautiful. It's also again, so complicated, I'm afraid I'm going to mess it up, so I've been delaying covering this formally. But basically what these layers are, the ectoderm, the endoderm, and not shown here is the mesoderm, are the layers, distinct layers of cells that will become fully differentiated cells. So if you see here the endoderm, here in green, these cells in this early stage of the embryo will become tissue like lung, like thyroid, like your digestive cells. And then if we go to the ectoderm, the blue layer, these are the cells that will eventually become things like skin, neurons, and pigment. So how you go from sperm and egg to a whole living organism is something that I'm really, really excited to talk about a little more, but I just want to tease a little bit of that to show off some of the immediately accessible ways that that process is very interesting. And here's where I'm going to have a little bit of chat interaction, because you're actually going to help hypothesize some of this, which is going to be very interesting. All right. Despite being so vital, and this is really, really surprising, really surprising, right? Despite being so vital, the process of embryogenesis, the process of forming the embryo, is not well conserved between species. It's not. And to me, as a biologist, this is really, really surprising. I want to give you a chance, chat, to tell me why this is surprising to me, or why it might be surprising to you. Like, the way that a human embryo develops, when you say not well conserved, that means not similar between species, right? Yeah, exactly. The way that a mouse develops is very different than the way that a human develops. It's similar enough where we can still like figure things out, but it's also not that similar at all. And so, the details on this process are actually quite different between species to species, which to me is very surprising. Why might that be surprising? And the clue is kind of in how I set this question up. Yeah, if we're all related, then why is it different? Exactly, right? Like, if this process is so important, you would think that evolution would have pruned our developmental process to be this hyper efficient way that always works every single time, and yet it is quite different. And I'm actually going to show you one other organism that is loved by developmental biologists and compare that to how a human develops, and it's absolutely insane how different it is. So many other aspects of our fully developed processes are highly conserved. Why on earth would something as foundational as going from the embryo to full organism be different? Exactly. How is symmetry broken in the beginning stages of embryogenesis? Yes, Mike W. Mike W, were you the microbio person in the other trivia stream? I think I owe chat 5 gifted subs, actually. I'll probably do that at the end of stream because you had surprised me with you getting that question from before, but that is exactly where I am heading to. But how are they different? Are the processes different? The chemicals? Oh, we're about to get right into it. Zebrafish incoming. I didn't actually choose zebrafish because I think I did pretty poorly in the zebrafish chapter. I picked Drosophila, actually. Okay, so this is a Drosophila embryo. So Drosophila, for those of you who don't know, is the common housefly. And Drosophila have been a beloved gift to developmental biologists because it is very, very easy to, I shouldn't say easy, straightforward to study. Is it that commonly in biology there are methods and proteins that can just be slightly changed to modify a cell, but for an embryo there isn't a well-defined system like DNA? The embryo does follow all the same rules as all other biological systems do, but the fact of the matter is that the strategies by which nature has imposed on how the embryo develops is surprisingly different. I just started watching the stream. Is this like a game? Yeah, it's trivia. We're playing trivia. Working on animating a Drosophila helicase literally right now. That's very exciting. This looks like the sweet potato I roasted yesterday. Well, I hope your sweet potato doesn't look like what I'm about to show. So this is an animation. I'll play it a couple of times. And I'm not going to give it any kind of context for now. And if you've seen my shorts, you've actually seen this video before. But let's just watch this. This is the Drosophila embryo, and each one of these purple dots is a cell, or at least a nucleus, I should say. And we'll see this in action. And yes, the GPU being able to show this animation is so exciting. It's awesome. Yeah, I hope your sweet potato did not look like that. I hope it didn't move that often either. So we'll play this video a couple times just to really, really drive home how insane this process is. My toilet brush, exactly. Creepy, I know. It really is. It's kind of interesting how different people come away with different things watching this process happen. But you can see in coordination, all of those nuclei dividing together, right? We go from a couple of huge nuclei, and then we see all of this splitting happen. And then all of a sudden there's this huge coordinated dance that happens as all the cells migrate to the places where they need to be. And this process is called gastrulation. Sorry, it's a little hard to say. Beautiful. Yeah, I wouldn't eat that. Neither would I. A little big for a neutrophil. All right, so let's watch it one more time. Okay, so here we have the cells on the outside, individual nuclei in purple, and what's fascinating to watch is the fact that these nuclei are all synced up. Isn't that kind of curious? I know I find that quite curious because you would think that just statistically that they would all be dividing at different times, but in order for this embryo to develop correctly, you need all these nuclei to somehow understand when they should be splitting. So we'll watch this coordinated dance happen, and we'll watch it for a couple cycles. Right, that's a cycle, right? They just all knew to do it, and we can even see that there are a whole bunch that are in the process of splitting their chromosomes. Bang, it's another cycle. They all do it all together in this synchronous motion, which is insane. And then here we go. That's another one. Whoops. Okay. And then in gastrulation, we need to move these nuclei to the correct place, and then that's where this coordinated motion happens, which is just mesmerizing to watch, just completely insane. How do they know when to do stuff? Exactly. And that's the part where I learned it in graduate school, but I'm not confident enough to recapitulate it for you because it is so complicated. But I remember just like learning how embryos develop and being completely blown away. It's so cool. I bet it's a chemical signal. Yes, it is a chemical signal. How that chemical signal propagates throughout this embryo is interesting and maybe not expected, which is very, very cool. Wait, it's fast, but you can still see the propagation happen like a wave. That's pretty cool. Yes, waves are a big thing in embryonic development. In fact, I was working on a video about Dunsparce, which I don't think I'll do anymore because it's so hard. You know that Dunsparce has evolutions, right? The Dunsparce. And that Dunsparce has either 2 or 3 segments in their body. And I wanted to do a video on the biology of dunsparce, and part of that mechanism by which you can form segments is wave dependent, which is very, very, very cool. I love this quality of stream so much better. I know, I'm so happy. Cell cycle, yes, that is what we are talking about. So, here's the thing. Chat, how does a human embryo form? So let's start by recapping a little bit about what you know about how an embryo forms in a human. And we'll go shortly after the deed is done, we'll say. So chat, tell me, what do you know about how humans develop? How do humans develop? A stork. I don't think I can formally endorse that hypothesis as a biology teacher, but certainly there are people who believe that, people being children. It's magic. Well, you could describe it as a magical experience if you want to romanticize it beyond biology. Sperm plus egg equals baby. We are missing a couple of steps between sperm plus egg equals baby. Yes, chat. You are all giving correct explanations for a very specific audience. A human develops within VS Code. Different context, maybe. Different context. There's like a bird and a bee. So where's the stork come in? They get downloaded from GitHub. Downloaded from GitHub. I like that one. Egg attaches to the wall. Chromosomal fusion. Yeah, 2 separate pairs of 23. Sperm plus egg equals zygote. Yes. Okay, we'll start there. Thank you, Kevin. Sperm plus egg equals zygote. What happens after zygote? What happens after zygote? What happens immediately after the zygote forms? So for context, the sperm enters the egg, and then at the point by which the sperm enters the egg, it releases a wave of calcium all over the egg to block out the other sperm. And then you form what is called a zygote when those chromosomes end up meeting. But what happens after zygotes? They start with one cell that splits into 2, and then travels and continues to split and travels until it attaches to the wall. Exactly. So you start with a zygote, and then the zygote splits into 2. Now this is very convenient to teach biology students because that just kind of makes sense. But for Drosophila, when you have fertilization, the sperm enters the egg, and then the nuclei start dividing, and they start dividing like crazy. So unfortunately, this video already has, I think, the cell stage where they've already split. But instead of going from 1 cell to 2 cell, the Drosophila embryo starts as 1 cell with like 100 nuclei at some point, 100, whatever the power of 2 is. And immediately that's so different. Right, that's insane, right? Zygote in humans, that just splits and then it splits continually. That's very easy to understand conceptually. In Drosophila, it stays as one big cell with hundreds of, or at least dozens of nuclei before they start to form membranes and like form actual cells. And that's what I mean by between species, there's surprisingly little conservation. This process is so important for every living being, and yet each one of us developed a way to develop an embryo slightly differently. And the fact that we are that different than Drosophila and right off the bat is just insane. I think that's so cool. Yeah. That is human. Doesn't the fly do it outside the body? I'm not entirely sure what you mean by that. So it's technically a unicellular organism in the video. So I think in the video, this frame starts after the cells have already formed. But if we were to rewind this video, I don't know how many minutes you have to go back, but you would see that it's just one big bean with a lot of nuclei, and that's just nuts. Insects are very, very far away from us in the evolutionary tree, though that's technically true, but we do study a lot about our own development by first studying it in Drosophila first. And again, the fact that this is so important, it surprises me how poorly conserved it is. And to your point, Megacraft, mice develop quite differently than humans as well, which is very surprising because, you know, we're not that different from rodents. I mean, we are, but we're not that different from rodents. All right. So this is the big question that developmental biology has instilled a deep love. This sentence is so screwed up. This question here, how does a cell know its own fate, is what makes developmental biology so fascinating for me. So chat, I want to get some hypotheses from you. How does a cell know what it is supposed to be? Or if we talk about my own neutrophil history, how does a progenitor cell know to become a neutrophil? You can speak very broadly, but if you start off with an egg and a sperm, how does each cell know eventually what it will become? Genes, that is certainly part of the correct answer. Everything does go back to genes. They draw straws. I do like that explanation. Maternal mRNA, ooh, Robert Furlong, an actual biologist it sounds like, but maybe we'll get to that in a little bit. Something epigenetic, something. Not quite, I think, although I can't rule it out. Oh my, I love how responsive chat is now with the Phi desktop. It's awesome. You can actually hear me in real time. That's awesome. It doesn't know until it gets the right mix of signals at the exact right moment during development. That's definitely true. But I'm looking for a bit more of a concrete kind of example. Maternal RNA is part of that answer. Signaling molecules, history of epigenetics, silencing. I believe that is true, although I have to reach further back in my developmental knowledge to confirm the epigenetic part. I think, yeah, there are genes that are silenced, but you are evoking memories of graduate school that I've since repressed. Yeah. Localization of growth factors. Hello, Otto. Yeah. Localization. Location. Yes, it's a fascinating question because differentiation is kind of positional too. Yes, we're all hovering the answer that I want to get out of this. Not to say that, chat, that you're wrong. A lot of you, I think, are saying correct things, but I'm not an expert in developmental biology, and it's been quite a long time since I've taken this class. But for me, what's striking about this process is the fact that it is location, and I think that is really, really interesting. You don't want some random cell deciding it's going to be a foot. It has to be in the right spot. Exactly. And we're going to talk a little bit about hypothesizing how a cell early on in development can get that location signal. So there's this concept called cell polarity, and that is referring to something similar that Mike's saying, symmetry breaking. The distribution of stuff nearby, it's on the tip of my tongue. Yes, very, very true. Symmetry breaking. So there's this concept called cell polarity. And this is something that I want, and it's been teased by some very smart chatters in the chat. Chatters who I think have taken as much or more biology than me, by the way, from given some of the answers that were in there. So cell biology is, cell polarity is the concept by which a cell can tell its own head from its own tail, right? So if we think about just the egg and the sperm and it comes together in a zygote, how do you think cell polarity is established? How can we start breaking symmetry to start being able to give the cell some positional information? What is the earliest, I shouldn't say earliest. What is one way that a growing human embryo can break symmetry and establish a sidedness? Chat. What's one way? Egg side versus sperm side? That is definitely one possible answer. Gravity, that's also a great answer. I think gravity is the correct answer for certain organisms. And again, it's been a while since I've taken these classes. This is the leftover egg cells that were not chosen. That is also true. There's some symmetry breaking even before fertilization. Are cells completely directional? Not always. Sometimes they need a little bit of symmetry breaking so that we can tell our head from our tail. But sperm entering egg, that is, in fact, one way that a cell can establish polarity. Magnetism, hypothetically true, and I don't know enough about different organisms to be able to tell you whether or not magnetism does play a role in certain organisms, but it's a good idea. Very, very cool. Wall of the uterus, that's another excellent example of a way that cell polarity can be established, right? If you are a dividing embryo and you're traveling down the female reproductive tract and you are looking to implant, the site of implantation provides positional information. It can inform whether or not, it can. I won't say that it does, because again, it's been a while since I studied any of this stuff. But once you start thinking about cell polarity, developmental biology becomes so much more interesting. This question of how do cells understand their heads from their tails all of a sudden becomes a very interesting geometric puzzle. Not only that, but cell polarity and this idea of development also contains a lot of overlap with things like cancer biology, and because cell polarity is a very important aspect by which cells determine their eventual cell fate, and so I love this question of cell polarity. It's so cool how cells understand how to arrange themselves or what fate to arrive at, which is very, very neat. Very, very cool. Gravity, I think, might play a role in the frog, but I'm not 100% sure, because it's been a long time since I thought about frogs. Yeah, very, very cool. And I can't wait to actually do the developmental biology journal club. But that's going to be one of the last things to do, because developmental biology requires you to hold so much information in your head at the same time. I really need to do everything before that first, but I just find the whole field to be really, really beautiful. Because when you watch this video, just to wax poetic, some people might look at this and think that it's creepy, and I absolutely understand why, right? Like, there's a lot of like little circles which might evoke a certain fear, right? But when I look at this, I feel a kind of kinship. Even though Drosophila develop way differently than humans or neutrophils do, there's a kind of elegance. Like, this is the way that we come to be, I'll say. It's revealing a little bit about our biological history, right? It's like a piece of our past, and learning how a fetus develops is like uncovering a little bit of your own personal past, a past that you were not smart enough, neurologically developed enough to witness yourself. And so to be able to view yourself in this kind of hindsight, I think is really, really fun. Can't fathom looking at this and finding it creepy. Way too deep in the bio pipeline. Yeah. I know, I feel that. All right. So that was my love letter to developmental biology. Let's talk a little bit about the hard questions. Where's a good place to learn this? I'm sure Khan Academy has stuff. If you actually want to learn stuff, Khan Academy is great. So let's do this question. Let me throw up the poll. Which molecule is the body's main neurotransmitter? Glutamate, dopamine, acetylcholine, or serotonin? So this is more specialized. So it's not hard if you've studied neuroscience at all. It's pretty easy. But if you're coming from basic biology, then this is definitely not easy. They are all important, but one of these molecules is the main neurotransmitter. The main excitatory neurotransmitter. I should have made that clear. Excitatory neurotransmitter. Which of these is more important than the others? Don't answer in chat. Let people come to their own conclusion. Of course, by virtue of answering, you are training your own brain. It's not important that you get it correct. What's important is that you try. They all have different functions. That is true, but there is one of these that is the main one. Come on, Dr. House, don't fail me now. Yeah, I learned a lot about biology through or I shouldn't say that. I learned a lot about disease from Dr. House. Ah, I forgot to start the timer. Okay, you know what, chat? You can have a little bit of time. You can have a little bit of extra time. You can have just that little bit of time. So those of you who don't know, my background is actually in neuroimmunology. So these hard questions are going to be pulling from the specialty knowledge that I have. So not my basic biology knowledge, but the specialty knowledge. Yeah, I'm gonna go ahead and hydrate. right. Okay, so let's go ahead and let's see what chat said. All right, chat, what did you say? Okay, the answer that chat has chosen is C, acetylcholine. That is interesting. Acetylcholine is the main neurotransmitter that muscles use. But there's a more correct answer for the body as a whole, and that is glutamate. It's A. It is A. It is glutamate. This is really, really interesting that the main neurotransmitter that when received by a receiving neuron evokes activity. And the thing is that we focus so much on neurotransmitters like dopamine and serotonin because these are important for the human experience, right? Dopamine, reward, learning. By the way, if you haven't watched my stream collab with Dr. Nils Wendel, I collabed with a real psychiatrist to talk about dopamine's role in surprise learning. Dopamine and serotonin often hog the spotlight as being things that are more relevant for our lived experience as human beings, but actually the workhorse of neurons is glutamate. That's the main way neurons talk to each other, which is pretty interesting. Didn't even know glutamate was a neurotransmitter. I know, it's kind of crazy how far under the hood glutamate goes when it comes to general understanding of neuroscience. Now you might be wondering, glutamate sounds kind of familiar, right? Glutamate is not something that you only hear in the context of neurotransmitters, but if you are a fan of Uncle Roger, you know exactly what this is. Glutamate, it's the same glutamate like in MSG. Now don't worry, if you eat MSG, it's not going to change your neurotransmission. That's not how these things work. Your brain is a different area than your stomach. But it's interesting that this amino acid can act as a neurotransmitter, and not only act as a neurotransmitter, but act as the main excitatory neurotransmitter that your brain uses, which is kind of nuts. Yes, it's the umami stuff. Yes, it is the secret to all good, tasty cooking, not just Chinese cuisine. Any kind of cuisine that you want to make taste more savory, add a big old pinch of MSG and you will be surprised at how much more eatable your cooking becomes. Yummy, yummy neurotransmitters. Exactly. Welcome, Sai. Welcome to the stream. Yeah, so that is very cool. I don't have too much more to say about that, but we'll go into the last hard question. So which of these molecules does the pufferfish neurotoxin tetrodotoxin TTX work on? GABA is the opposite. That is correct, Chad. So I'll go ahead and actually start the timer this time, the timer that you unfortunately can't see because of the scuff. Pufferfish poison, a very, very near and dear subject to my heart because it is one of the first projects that I ever took on as a student. In the 5th grade, we had this like open project where we could study anything in science and make a book report on it. And like a little freak, I chose one of the deadliest toxins to ever exist. And so that is the tetrodotoxin. The pufferfish poison comes from the pufferfish's liver, very, very deadly. And I'm wondering if you know which one of these neurotransmitters TTX actually works on. Glutamate updates synaptic connectivity. Yeah, so glutamate does play a role in adjusting the strengths by which neurons communicate with each other. GABA is in fact the opposite. Careful with adding more sodium to your meals though. Secretly salt. Yeah, that's true. It's true. If you are someone who needs to keep track of their salt levels, that is true. Flavor comes at a price. All right, it is a neurotoxin. So which of these receptors does the poison act on? Poison, tasty fish. Yes, fugu. You can have some fugu in Japan, pufferfish. I actually have never had fugu, even though I'm such an adventurous eater, just out of principle, because it's kind of an insane way to go. You essentially suffocate, but you are awake for every moment of that. So in no world is that worth it for me. That's one that I am going to continue to avoid. All right. Okay, so chat, let's see what you said. So chat chose acetylcholine B. So acetylcholine is the main neurotransmitter for muscles. And you would think that that is the case. You would think that that would be the thing that TTX blocks. But the thing is that it's actually way scarier. TTX blocks something that is way scarier than acetylcholine. At least to me, as a person who has studied neuroscience. And the reason why the real answer is much scarier is because TTX actually blocks sodium channels. Sodium channels. Chat. If you know the answer, don't respond, because this is, you know, if you know the answer, then you know. But we'll try to give people that have taken biology maybe a long time ago a chance to respond. Why might blocking sodium channels? Why might blocking sodium channels be scarier than acetylcholine? I mean, scarier is subjective, but it scares the crap out of me. What does sodium channels do in the body? It's gotta be acetylcholine sometime. Keep it picking your chat, true. Hello, Toxo, welcome. What do sodium channels do? Where have we heard of sodium channels? This should be, I think, in bio, AP Bio, maybe. Sodium channels. Okay, so we've given people the time to think. If you know the answer, what is the answer? Something with sodium, potassium. Yes, okay, we're getting there, we're getting them. Sodium is important for keeping your cells whole. That is also true, but what about it being a neurotoxin? Muscle contraction, that is around the correct answer. Charge potential, getting closer. Neuron firing, thank you, Psyche May. Welcome, Psyche May. Very good. Required for action potentials. Thank you. Action potentials. So let's talk about action potentials. Yes, thank you so much. Tetradetoxin blocks action potentials. So what is an action potential? Action potentials are ways that neurons communicate with one another. So we see here in this animation, a electric potential traveling down the axon of a neuron. So the cell, the neuron, keeps a charge difference between the insides and the outsides. So the inside is relatively negative and the outside is relatively positive, with a lot of positive ions like sodium, calcium, potassium on the outside, and a relatively lower level of those ions on the inside. In order to pass off an electric signal, this wave of propagating difference in charge has to make it from the top of the body to the end of the body, right? And so the way that this happens is through what is called the action potential. And what this involves is a bunch of sodium and potassium channels opening and closing at certain times, allowing positive ions to flow in and then eventually flow back out. And this wave of propagating electric signal, you can call, will eventually make it to the end of that neuron, and that change in charge will trigger the synaptic vesicles in the end of the neuron to release their neurotransmitters for the next neuron to take up. And so TTX blocks action potential. So TTX blocks everything that a neuron does because it can't propagate the information downstream. Your neuron is frozen essentially by TTX. They're completely quiet. And that's insane. Right. So when we talk about acetylcholine, yes, acetylcholine, that would do it, right? If you can't contract muscles. Although I'm not entirely sure if smooth muscles like the lung work differently, because my physiology isn't that good. But TTX will shut up any neuron because it can't do action potentials anymore. No neurotransmitter, it's not specific. It'll just make all of your neurons really quiet. Wire blocked. Heart no get pacemaker signal, exactly. Diaphragm no get signal breathe. Yeah, yep, Clockwork's got it right, exactly. That's terrifying, right? It's not that it's blocking muscle contraction, it's blocking neurons from communicating at all, and that's really scary. 13 years since I took biopsych, still got, yeah, you know, way to show that off. It is terrifying. It is terrifying. Could it get into the brain? Does it shut down your neurons? I don't know enough about the pathophysiology of TTX poisoning. But I will say that TTX was a tool. I actually got to work with this poison. It was insane. My thesis advisor used to say that we had enough neurotoxin in our freezer to kill everybody in our state. It is so deadly. You need so little of it to completely kill, you know, a large population. It's bonkers how little of this you need, because again, it just stops your neurons from communicating. I guess the first to go in this metabolic pathway would be the lungs. I think the blood-brain barrier would stop it. I'm not actually sure that it would. I can't say that I know that for a fact. No wonder you sworn off eating food. Exactly. Let me tell you, one of the most terrifying days of work was when I first had to use this neurotoxin. It was really, really scary. I kept on, like, I don't know, my heart was beating really, really fast. I was about to have like an anxiety attack because, you know, you chip pipette tips. If you cut yourself shallowly, maybe you introduce a little bit of it into your bloodstream and then you just stop breathing, and if you're alone in the lab after hours, you just die where there's nobody around. Like, that's no good, right? Of course you have to abide by very strict safety regulations in order to work with stuff like this. Yeah. What is the blood-brain barrier? There is a barrier between the brain and the rest of the body. Helps keep the brain free of a lot of the stuff that will hurt the rest of your body. All right. So what was I going to say? Yeah, I used this. And what did I use this for? I used this particular poison to quiet neurons. So it turns out when you add TTX to neurons, they stop firing action potentials, right? They get really, really, really quiet, because they're deaf, right? They can't receive any information, they can't propagate that information, and so networks go quiet. But it turns out, and this is something that you would only know with very specialty knowledge, is that even completely poisoned neurons that are not doing action potentials at all are still capable of passing signal. Sorry about that. They're completely quiet in terms of they don't fire action potentials, but they bleb little vesicles of neurotransmitters, these mini excitatory postsynaptic potentials, these little tiny little synaptic packets that get released on, you could call it on their own, that are not dependent on action potentials. And this is a measure of synaptic strength. So it's sort of like your baseline firing, which I think is really cool. I didn't know that even through TTX, your neurons still can communicate. It's just a lot quieter, and it's not dependent on the axon, which is insane to me. Very, very cool. They gesture suggestively. Yeah, you know, Tyrannoteer, sorry, I can't pronounce your name. Tyrannoteer. That's actually a gag that I have coming up in the Prion video. You've kind of predicted one of the jokes that I put in. Neurons sharing secret messages to one another in their own DMs. Very, very fun. Twerking neurons. I'm not that good at animating to do that, but maybe one day. How much is necessary to kill a normal person, and does it matter how, where, or how it enters the body? It's a question for a toxicologist to answer. I don't know myself. Maybe TTX isn't 100% efficient in preventing action potential. It kind of is. It's really, really good at blocking action potentials, like really, really good. It's this completely other independent phenomenon that is just a part of the basic biology of a neuron that lets it bleb out the occasional blip of information. And it's different than an action potential. Maybe one day I'll get into it. It's a very, very niche field of neuroscience. I'm imagining a single dynein running along the axons. It wouldn't even be that. It's actually just, see at this end here, the presynaptic dendrites, synapses. Down here, there's actually just machinery that occasionally blebs out neurotransmitter. It's pretty interesting. Okay, chat. That is 4 out of 5. 4 out of 6 that you got right. So let's write it in here. 4 out of 6. Let's see how much. Yeah, if I do as well as you on the trivia that you all submitted. So if you didn't know, you can participate in this process by submitting questions to me. Now, I haven't looked at any of the answers. I have all the answers open in my Google form, and I'll consult them as I do this. But you're free to play along, so I'll go ahead and start a poll for this one. So this is, and I just did this in order, so if I don't get to your question, it'll probably be in the next stream, the next trivia stream. So this question was submitted by, I wasn't collecting names at the time, which is unfortunate, so sorry I could not give you credit, but this is a pretty neat question. What was used to treat syphilis before the invention of antibiotics? Hydrogen peroxide, calomel, rotten cheese, or quinine? These are all really fun answers. And, you know, I'm not going to look at chat because I want to answer this very honestly. Syphilis being treated with hydrogen peroxide doesn't sound feasible to me because applying hydrogen peroxide to your regions, I think, is kind of insane. So I'm going to rule out A. I don't know what calomel is, so I'll leave that on there. Rotten cheese, I mean, it could be a reference to something like penicillin, but I doubt that. And then quinine. Quinine sounds like a modern day antibiotic, so I don't think it's quinine. So I'm gonna go with calomel by process of elimination. And I'm going to end the poll for chat as well. And then I'm going to check my Google form, which I closed out of. I can never take the scuff out of the VTuber. Give me just a moment to load up those answers. Alright, the answer is calomel. Calomel is a mercury derivative and was used to treat syphilis before modern antibiotics. Okay. Okay, that makes sense to me because of the way that these others were worded. I think hydrogen peroxide, I was tempted to say hydrogen peroxide. I was tempted to say hydrogen peroxide because that just sounds like something that old-timey doctors would do, right? Just rub hydrogen peroxide on nether regions, but I think it's a little too crazy for me. So I'm glad to have gotten that one right. What does chat say? Calomel has mercury. How do you all know what calomel is? I've never heard of this before. It's in tonic water? If you drink cocktails, you probably ingested some. Calomel or mercury? I didn't know that. Oh, quinine is for malaria. Interesting. Interesting. Modern medicine got really wild between the 1850s and the 1950s. Yeah, I wouldn't really know. And that's why I was very tempted to pick hydrogen peroxide. All right, next question. What is the... Oh, God, okay. What is the lightest element, lowest proton number, which has no stable isotope? Chemistry is not my strong suit. And again, at the time I wasn't collecting names, so I'm very sorry about that. I'm not looking at chat, but I'm going to talk out loud. So, the bigger the element is, the less stable it is at a certain point, because there's that thing that's like the island of stability or whatever, where there's like, no, that's wrong. God. I'm gonna make myself look like a fool. It's been quite some time since I've taken chemistry. I bet Toxin would know the answer to this. Okay, 21 seems kind of low. Uranium is a safe bet for things that decay. Oh God. I don't know, because things like carbon-14, which is an isotope of carbon, do decay. And that's a very low proton number. I think, okay, well, if it's carbon-14, if carbon-14 is not stable, that means that there is basically no lower limit here, so I can't really eliminate any of these. I'll eliminate uranium because that seems like the bait answer, which leaves me with a 1 in 3 shot of guessing this completely correctly. I'm gonna roll a 1d3 and allow that to determine my fate. So let's roll a 1d3. and I rolled a 1, so I will say scandium. My very scientific approach, scandium. The answer here is technetium 43. By the way, this person who submitted this said this was easy. I don't think this one was easy. So chat, if you had picked C, you are correct. What did chat say? 43% of chat got it. Wow, okay. I did not get that right. I had no idea. I'm gonna read chat for a bit. Chem, easy, totally not. Oh, Engine Crafter, this was your question. This was not easy, man. This is not easy. Multiples of 7? Oh, is that what this is referring to? Dang, what the heck? Took chem this year and don't remember. It's been a while since I've taken chem. Has no stable isotope. Carbon has a stable isotope. In any case, I would not have known it either way. I can't believe that chat got that. That's kind of nuts. That's nuts. This is Engine's revenge for that medium question from earlier. True. Yeah, there's no way I would have gotten that. You totally trounced me. Okay. Very cool. Very cool. Chemistry question for a biology channel. Yeah, I allowed anybody to submit any science question. Although if you wanted to do biology, the thing is it's very easy to pick biology questions that I would not get because you can get that specific. So I figured this would be fair in the sense where... Actually, I don't know. Chat, what do you think? Should we do a pure biology trivia where you challenge me with biology trivia? That could be kind of fun. Let's start that poll. Pure bio trivia one day. We'll leave that to the chat. Yeah, because like I don't claim to be some sort of biology super genius, right? I have a notoriously bad memory. But that could actually, and that was the original reason why I wanted to make this all science, because I feel like that would be a little more inclusive. Anybody could ask questions based on their own scientific specialty. But now that I think about it, because I'm not that confident in my ability to know all of biology, it could be a fun way for you to all trounce me in my own field. Gotta ask my bio friends for some question tips. Chat needs a fighting chance to beat 5. I don't know about that. Chat has done quite well. Again, my memory about biology is not very good. I'm sure you could all come up with biology questions I would not be able to get on the fly. Revenge, yeah, exactly. Beat me at my own game. So it seems like most of you, maybe what we'll do, because chat is a little split, is we'll have the bio versus bio part, and then we'll just do other sciences just as a fun round at the end. I think that might be a fun way to do trivia. That might be fun. Yeah, because I do want to test my metal. You know, I've been talking all of this guff about testing your own metal and how that makes you learn, and it's about to test my metal too. How closely would it have to be to biology? I would prefer to stay in the realm of cell and molecular biology, because that would actually have personal stakes for me, right? Like if you asked me a question about birds or something, well, I'm not a bird scientist, so there's likely no way I'm going to be able to get that. But if you ask me something about the developing Drosophila embryo, that's something that I did learn at some point, and if I were to get that wrong, it'd be hugely humiliating. So as a VTuber, it is my job to deface myself for the pleasure of my audience. All right, yeah, histology, you can definitely ask something about histology. Actually, can you? I don't really think I learned histology formally, but you know, it's more in the realm than like something about birds. It would be more satisfying if you managed to beat 5 in biology. Exactly. It'd be harder, but I'm sure you could do it pretty easily. The thing is I can ask myself questions that I would not be able to get. How good am I at paleontology? Not good at all, but I don't really count that in cell molecular. Okay, let's go on to the next question. What was the first discovered virus? What was the first discovered virus? Okay, so rabies was one of the first developed vaccines. I don't know if it was I don't think it was the first, right? Because that one's polio. But it's one of the first was rabies. I think rabies and polio are up there as traps. HPV, I don't think that that would be easy to isolate because it is specific. I actually don't. I think. Oh man, if my fellow grad students were here, they would absolutely be roasting me because a lot of them did work on HPV. I think diseases that are transmitted through intercourse are probably harder to culture because they tend not to like being they tend not to like infecting cells that aren't exactly the cells they need to infect. So tobacco mosaic virus, I think, to me sounds the most likely, just because it's far easier, especially if we're talking about the past, far easier to culture a plant than it is to culture mammalian cells. So I'm going to say C. C is going to be my answer. Okay, and then this will end it for chat here. Chat says poliovirus. I mean, maybe. First discovered virus. Let's see what the answer is. So this one actually has stakes because, you know, I learned about viruses, significant part of my education. Yes, tobacco mosaic virus was first to be isolated and discovered. Before, people used to theorize that virus molecules were some sort of toxin produced by bacteria. Oh, that feels good to get. My brand remains intact. Oh, that's good. Okay. What was the reaction to that question? Let's see. Smallpox is the first. Is smallpox the first? Oh, smallpox. Oh, the vaccine question. Right. Oh, I'm glad that that question wasn't about the first vaccine then, because I would have gotten that wrong. That's good. Any grad student should understand. Brain too full, things fell out. Yeah. Yeah, tobacco mosaic virus. Oh, it was split between chat. 47%, 47%. Yeah, chat, you get half a point for that, I think. Chat was split. Yeah, because I was thinking, you know, viruses are very hard to image, especially back in the day, and so you probably needed a whole lot of it to actually see it. And the way that you would see something like that, it would be easiest to grow it in a plant. I don't know if that's actually why it's the tobacco mosaic virus, but it certainly makes sense to me. Okay, let's go to the next question. How many chromosomes do non-human apes have? This was submitted by Persica, who is another VTuber who does, what is it? Anthropology and primatology. Very cool. So you can check her out at Persica Arts on Twitter. So we'll start this question. Oh, and here we go. How many chromosomes do non-human apes have? 48, 32, 46, or 74? So I am going to say that non-human apes are probably close enough to us in evolutionary time for it to be roughly where we are at. So if we are 46, then I'm going to say A. I think, you know, I don't think this is a bait question. We should be pretty similar. So what is the answer here? Persica, did you fool me? No, 48. 48 is the right answer. Oh, I forgot to end it for chat. Ooh, chat picked C. Oh, 46 would be the human number. You can remember this by remembering the company 23andMe and then just multiplying that by 2 for humans, right? Yeah, non-human apes should be a little different. So let's read the answer that Persica gave us. 48. Despite being so similar to us genetically, apes show a plethora of biodiversity. In fact, 2 chimps from nearby populations possess more genetic diversity than 2 humans from separate continents. That's really, really interesting. Okay, yeah, no, that feels good. That's another biology question that allows me to protect my brand. Yeah, because I think I remember learning this formally at some point, but it's good to know that has been retained. Yeah. Had to bail for a bit. No worries, entering dad mode. True. Glad I don't have to deal with that right now. It tickles me that I have to qualify non-human apes. Well, you know, it's important that we acknowledge who our real cousins are. This is really fun. I love trivia night. I think this is a lot of fun. I love these questions, and I love getting to talk about science. It's just so great. Okay. When is the estimated release date for the Prion video? I want to release it by the 15th. No promises there, but that's about when the video should come out. About when. Okay. Let's go to the next question. That was a good question. That one, I was pretty confident I was right, but I was actually, if I had looked at chat first, maybe I would have strayed from the true path. This is an interesting one. Which of the following foods do not typically involve the Maillard reaction? Let's go ahead and start the poll there. Which of the following foods do not typically involve the Maillard reaction? So the answers are grilling steak, roasting coffee, toasting bread, or D, none of the above. So the Maillard reaction, I believe, is a chemical reaction that forms browning. So it's a chemical reaction that imparts a good amount of flavor, and it is the result, I believe, of turning protein, denaturing protein into different proteins. Because for sugar, that's caramelization. So where I am split here is, I think grilling steak is definitely Maillard reaction, right? That's definitely protein if it is protein, because I think Maillard reaction and caramelization are different. Roasting coffee, I'm also going to say, is probably protein-based. I don't know coffee to be heavy in starches. Toasting bread is where I'm kind of maybe potentially overthinking it, because bread is starchy, that's converting sugars to different sugars when they are browned. But I actually don't know if browning on bread is the result of protein or sugar being converted, or none of the above, which is, this is such a well-crafted question, because I'm split between toasting bread, because that browning could be caramelization, or none of the above. My guess is because the brown part of bread doesn't actually taste sweet, that is also a protein thing, because bread has gluten in it. Gluten are protein strands, and because the brown part of bread doesn't taste sweet, I'm going to guess that that's a protein thing and not a sugar thing. So my answer here is going to be D. Chat, what did you say? D by 37%. So there's a lot of non-consensus within chat, a lot of split reactions. Split between D and B, which is interesting. Okay, what is the answer? D, they all involve the Maillard reaction, which occurs at the high temperature involved in cooking proteins and sugars. Oh, cooking proteins and sugars to form more complex flavors and aromas that enhance the flavor of food. That is a good question. I love cooking content. I have a lot of my YouTube recommendation stuff is cooking content, because I am someone who derives a lot of pleasure from eating. I'm a professional phagocyte. What can I say, right? Eating is so much of what I enjoy. So I love watching like cooking content. It's not that I love watching cooking content, because I don't search it out. The YouTube algorithm knows that I will click on cooking content. I think that is the more accurate way to describe this. All right. Have I been pronouncing it incorrectly? Oh no. Maillard. Is it Maillard? I see, it's Maillard reaction. I see. Oh no, none of the above option. Yeah, exactly. All right. Totally not my question. Thank you for the question, Morjuice. That was a good one. Really tripped me up. How many questions are there? There's one more question. I tried to keep it even. 6 questions from me and 6 questions from chat. All right. Have a whole episode about the biochemistry that makes cabbage and other brassicas so incredibly delicious. Ooh, I could talk about hot pot strategy. Okay, here's some hot pot strategy, right? So I didn't read this paper, so I can't like determine whether or not it is very scientific or very robust, but there are papers out there that have determined combinations of different foods and different aromatics that produce a stronger flavor. So Napa cabbage and soy sauce have complementary savory components that go very well together, supposedly. And so if you are ever out eating hot pot, my strategy, and this is a strategy that I learned from Toxo, is to add Napa cabbage to your hot pot. You might be thinking, if you're eating hot pot, why would you not just fill up your whole thing with meat? And you could. If you are eating all you can eat and you want to get the bang for your buck, of course you eat meat. But if you want, I think, the absolute best flavor experience, you start with Napa cabbage, because Napa can donate so much more savory elements to your soup. It's really incredible comparing a hot pot that has had Napa added to it and a hot pot that has not. It's night and day. It's such a incredible ingredient. All right. Let's see. What's hot pot? It's also called shabu shabu if you've had that in your area. It's I'll call it broadly Asian. You could call it Mongolian because I think that's where it originated, but I'm not entirely sure. But basically, these are restaurant establishments where you have a boiling pot of broth and you can order things to put in said broth, and it's delicious and awesome. Hot pot is like barbecue, but instead of a grill, you have hot soup. Yeah, that's a pretty good way to describe that. Yeah, I love this question. It's made me think very fondly about all the cooking content that I've consumed over the years. Okay, next question. Last question. So I am 1 down. Chat, you are 2 down from my question. So if I get this, then I win this round of trivia. If I don't get this, then we are tied. Yeah, okay. Next question. This is the final question. Which of the following is a way to decaffeinate coffee beans? So we have coffee appearing in the trivia once again. So I'll go ahead and set that up. This one's also by Anonymous. Okay, which is the following way to decaffeinate coffee beans? This actually really screws with me because I know you can decaffeinate coffee beans with different solvents, which is why decaf coffee tastes kind of bad because of the solvation process. So adding chemicals that can extract the caffeine molecule, but that process can also ruin the taste of coffee. I know this because sometimes I will have decaf because I'm very sensitive to caffeine. But unfortunately, salvation is not one of these options, which kind of sucks. So this one, I think, is like a true guess on my part. But I'll talk through what I'm thinking about anyway. So, fermentation. Oh. Fermentation. I'll leave this one up because I think that caffeine can be broken down biologically by microorganisms. I think. I've never heard of fermented coffee bean before. Electrolysis, so submerging coffee beans in some kind of liquid and shooting current through them. I don't think caffeine is a charged molecule, so I don't think that you can get caffeine ions to stick to an electrode. Although I could be wrong on that, because I have a vague memory of what the caffeine... I didn't do any research on all these questions, which is why I'm so uncertain on, obviously, to keep it fair. But I think caffeine is a ring structure, and I don't think it is ionic. Like, I don't think there's a caffeine salt, so I don't think it's electrolysis. Water. Water would imply that you can just soak coffee beans and the caffeine will leach. Again, with the ring structure, I think caffeine is... And I could be wrong, and chat could be yelling at me right now, and I'm not looking at chat. But I don't think caffeine is tremendously water-soluble. Could be wrong. But if you leave coffee out for a while, it's not like you get a caffeine oil that forms on the top of the coffee if it were totally not soluble in water. I don't know. It doesn't feel like water. It feels like a waste of coffee beans. Vacuum chamber. I don't think this is correct because all that will do is potentially drive off water. I don't think caffeine would vaporize from the bean itself. Or could it? I don't know. It's really tough. I think I'm only eliminating electrolysis and then I'm down to fermentation, water, or vacuum chamber. I don't want to think too much more about that. I'll go with my gut and I'll say A. So A is my answer. This is a really tough question. Chat, what did you pick? Chat also picked A. Okay. Okay, we're locked in on A, folks. What is it? C. The Swiss water method of decaffeination is a chemical-free process that involves soaking coffee beans in water that is saturated in coffee compounds from a prior soak but has the caffeine filtered out. Thus, only caffeine can be extracted from the next batch of beans. That's insane. Yo, that's insane. What the heck? That's crazy. I had no idea. That's really clever. Technically, that's not just water though, so I'm gonna rate that question as eh. Because imagining coffee beans and just water is like, okay, well, it's just gonna leach everything equally. But if it's super saturated with coffee stuff minus caffeine, you still had to extract that caffeine out somehow, probably by solvation of some sort. So, I technically didn't get this right. I mean, fermentation is not the right answer, but between you and me, Chad, I think C is a little unfair. It's a little bit of an unfair answer, but I still love this question. It made me really, really think hard about the answers. Yeah, let's see what the chat was saying while I was thinking. Secret OChem question, what is caffeine soluble in? Don't remind me of OChem, please. Electrolysis does not sound very targeted. Yeah, that's why I was like, I don't think it's... it could be if caffeine was ionic, like a metal or something, and then it could attach to an electrode. But I don't think caffeine is a salt like that. I wish I was as insensitive to alcohol as I am to caffeine. It just means you're alcohol efficient. That's what my grad student friends used to joke about. I'm a very alcohol efficient individual, we'll say. I thought the normal process was filtering using water, but you still need to separate the caffeine and other soluble products. Yeah, exactly. That's what I'm referring to, the fact that water is maybe not the most fair answer here. If water wasn't water soluble, brewing coffee wouldn't work. Yeah, I corrected myself mid contemplation. Really good question. I love this one. But that does mean that we are tied, chat. 4 questions that you got right, 4 questions that I got right. That was awesome and a lot of fun. Yeah, really, really good. So if your question did not appear on the stream, that is because I wanted to keep it even, 6 and 6. I think this is a pretty good ratio of questions. 6 of my questions, 6 of yours. And it's made for a pretty lengthy stream, an hour and 40 minutes in, and which I think is like really good stream time. Thank you all for submitting your questions, and of course the next time I do trivia night, I will put out another Google form where you can submit more. So if your question was not here, it'll likely be in the next one. All right, thank you, y'all. Why don't we do just a little bit of chatting before we call it a stream, because it's been a little bit of time since I've had the chance to talk to you all. So let's go ahead and let's get a little bigger over here. All right, of course we gotta scale the chair too. All these assets were of course made by Toxo, which is super fun. I love this presentation background. Yeah, so let me tell you, chat. We almost got 5, chat. Almost. I'm sure you'll get it next time. We'll do more of an even split between biology and the bonus trivia, we'll call it at the end. Yeah, maybe we'll do bio versus bio, and then we'll both compete for the trivia questions as like the tiebreaker stuff, which is really neat. Tiebreak from Toxo. Toxo did not submit one this time around, but maybe next time. According to a Google search, fermentation does reduce caffeine content too. I know that there are microorganisms, especially fungi, that love caffeine. That's what I thought too. I think I should get half a point on that. I think I should get half a point on that. Although, Tyranitir, I think we have to check whether or not those coffee beans are fit for human consumption. Because plenty of things can break down caffeine, but I think the spirit of the question was asking in a form that that humans can consume. So this means Phi is as smart as 20 people. That's not really fair. Some of these were more in my wheelhouse, and I picked some very nasty biology questions for you all, but 4 out of 6 ain't bad. Chat did quite well. I think I only really managed to trip you all up on the very detailed, crunchy neuroscience stuff, which isn't even the draw of my channel, right? Like, I barely talk about neuroscience, a little bit in the rabies video, but one day I will be confident enough to get real deep into the stuff that my graduate research was in. Fermented coffee beans doesn't sound like it would taste like coffee anymore. I don't know. I love fermented products of all sorts, and I would be very willing to try fermented coffee. What are my thoughts on neutrophil extracellular traps and how do you think they evolved? I don't know how they evolved. I do think they are very cool. Of course, with great power comes great responsibility, right? Because a lot of the times the neutrophils do that, they are no longer operational, depending on the extent by which they throw out their net. They tried to make the acronym NET. I think that's called a backronym, which is really fun. Like an acronym that you want to make, and then you justify it retroactively, a backronym. What is my favorite dinosaur? It is the Velociraptor. I loved Velociraptor as a kid. Although, back in my day, they were still smooth-skinned, reptilian-looking badasses instead of weird birds. Although I still love them all the same nowadays. Crunchy neuroscience, yes. I think that the reason why I'm a little nervous to do the crunchy neuroscience is because there's quite a bit of background, quite a bit of context to try to get into. As you could tell from my collab with Dr. Wendell, the brain is really freaking complicated, and it's kind of difficult to make it accessible for an audience that hasn't had a little bit of training, but we'll get there, I think, just because I am someone at the molecule level, and of course, to understand the brain, you have to understand it at the cellular, the molecular level, the systems level. And I think because there are so many of you in my audience that are like computer people, I think we can collaborate, you, the audience, and me, on trying to figure out the brain, you know, because I have information on how the molecules and the cells work, but I don't really have the abstract understanding of how networks work quite as well. Dr. Wendell had to be very patient with me last stream, constantly correcting my assumptions based on how the brain is wired, because I focused very little on how the brain was wired back in my grad school days. Nerve-breaking brain jerky. It's one tissue that I will not eat. Again, being a professional phagocyte, I will eat a whole lot, but nervous tissue I will not get into. Again, same reason why I don't eat fugu, because of TTX. I don't eat nervous tissue because of prions, which you'll hear a lot more about around July the 15th. All right. I love fermented milk. Yeah, I mean, I think one of the most surprising foods that I've had is salted fermented milk or salted yogurt, which I would be very willing to try again, but the first time that I had it, it was in fact very shocking. What about the civet coffee? That is true. I wonder if the civet coffee is fermented or how fermented it actually is. Sorry, I'm catching up on chat. I realized, oh, it's so good having chat be like live. I'm actually like behind chat, not because of the lag, but because y'all are chatting. That's awesome. I'll jump ahead a little more. What organ system is the most fascinating to research? Of course, my answer is going to be the brain. That's what I did my research in, so. Thomas thermonuclear bomb. I think you have to, there's instructions in the rules section to fully activate your Discord membership. Decaffeination question, you mentioned solvent, and I seem to forget that water is a universal solvent. It's true, but for some reason my brain imagines caffeine as like an oily residue, but that's just like a weird assumption that I made out of nowhere. Just teach the entirety of neuroscience in the first half of the video and discuss the topic that you want to discuss. That is the general format of my streams, but the issue with that is that I'm very particular in the way that I like to teach. I like to build in thought-provoking questions because I think that is the more interesting way to learn. I could do a neuroscience dump and treat it as a very traditional lecture, but to me that isn't fun. I think it's more fun to have this back and forth where I challenge you, chat. And that's not always easy to set up with neuroscience because it can get kind of crunchy. I'm a software engineer. I was binge watching Chubby Mew the other day and realized his case studies are basically the same kind of stuff I do every day when I'm debugging stuff. Yeah, debugging, I think, is something that scientists and programmers have in common. Just want to say I really appreciate people like you who make various subjects accessible to people without academic training. Love this channel. Thank you so much. Yeah, thank you so much. I think I have a deep love of sharing cool information. That's why I got into teaching. Some teachers who teach students get into it because they love like students. And it's weird to say that I don't love students. It's not that I don't like my students, right? Of course not, right? Like I think that they're very capable, and I am excited to, oh, my chat, that's so weird. The chat was offset the entire time. I think that they're very capable, but in the structure of the stuff that I teach, I kind of get them for a little bit and then they disappear from my life forever, right? And so there's not really a chance to see their entire arc of their development over time. And so I view those teachers who like really love forming these like fully fleshed out people and improving their lives in meaningful ways. I respect them so much. My pure joy, like the thing that is my passion, is sharing cool things, and teaching happens to slot in very well with that. Of course, I care a lot about making sure my students come out of my class fully formed, well, as more fully formed intellectually than they were coming into it. But in terms of like, if I were to be fired, you know, the next day, I will find a way to talk about things that I find are cool, no matter what. Most of the time that's teaching. It's kind of an interesting way to think about why you like doing the things that you do, and I thought a lot about it over the course of my career, right? I love teaching, but that's because I love when like a student realizes something is cool for the first time. Not for the first time ever, but for the first time in my class, right? If I'm talking about a certain system in biology, and you see them like light up and they realize how cool it is too. It's like sharing what you find is cool is like always such a cool feeling. And then my intellectual passion is to make sure that they are fully equipped to think, which is very neat. As a psychologist, we're at least several centuries from understanding the brain, I swear. I know, it's really, really tough. But when you start to get a handle on how circuits are wired and how certain... So I'll say this, Psyche, I agree with you that there's so much that we don't know about the brain. But actually, there's also a lot that we do know about the brain, surprisingly. And I'll tease it a little bit here, but there are scientists who have isolated the circuit that controls some aspects of motor coordination in mice. And so what they can do is that by triggering these very specific neurons, they can make a mouse turn left or turn right with very high precision. They turn a switch on, the mouse turns left. Turn the switch on the other way, the mouse turns right. And that's incredibly... The insight that you would have to have on how neurons are wired, that's crazy. That's a crazy level of detail that we do understand, and there's a crazy level of detail that we don't understand. As a computer science, same, but with how complex artificial neural networks work. We actually have overlap there where I was learning a lot of like 101 machine learning stuff to try to understand how neurons work at a conceptual level. And that was actually quite useful in terms of a framework to understand the brain. What are other places to learn this stuff on an advanced level besides Khan Academy? My channel. Kirkazad, of course, has great content. SciShow has great content. But if you want deep stuff, you gotta go to college. Yeah. That's why you pay for a college education, although there are a lot of universities that have their lectures for free online. If you just search for free biology, like a lot of the Ivy Leagues actually have free courses. You just have to search for them if you want stuff at this detail. Do you have any other systems in the human body besides the nervous system that you have an interest in? Of course, I love immunology. I was never a formal immunologist because immunology is so crazy complicated, but the story of evolution is the most fascinating when you're talking about us versus germs, because we propel each other to evolve in the most direct way possible. It's literally a fight for our survival. And ecology, you know, evolution, ecology, adapting to the environment is also important. But in terms of when we're talking about molecular biology, like looking at the level of molecules, I think there's no more fascinating battle between the immune system and germs. don't see in the rules section. There's a emote that you have to react to, and once you react to that, you should have all the rules. Teaching is just info dumping the profession. Positive. Yeah, it's true. And actually, the info dumping part is among my least favorite aspects of teaching. I like teaching how to think. Thinking through an experimental problem, I think, is the most satisfying thing. And if you're a gamer, gamers out there, like the cognitive processes that you use for gaming, problem solving, it's the same in research, right? And getting to train people in the art of problem solving is really, really fun. And what's less fun is kind of, oh, the TCA cycle, the first input is acetyl-CoA. That stuff isn't as fun as like, how do we study the citric acid cycle? That'd be really, really neat. Just remembered in college, I spent a summer doing neurology research. My engineering professor was partnered with a neurosurgeon in researching deep brain stimulation for epilepsy treatment. Epilepsy is something that I have thought a lot about as well. Not only is it sort of something that I've seen in my personal life, but also it overlapped very heavily with my graduate student research. So epilepsy is also... brain injury in general, it's one of those things where I'll never make a video about it because it's so dark, although I'm making a pre-on video. But again, it's like the balance between levity, making things light, and treating things seriously is pretty hard to balance. But epilepsy is also, from a molecular level, interesting. I'm not going to say epilepsy itself is interesting because, you know, real people have epilepsy, but the mechanisms by which epilepsy occurs, I think, would be worthwhile talking about one day. Neural networks are the mathematicians and engineers finally getting a glimpse at how the rest of science can be. yeah, it's kind of a black box. It seems like complexity that our brains cannot handle. Like, our brains are so complicated, but our minds are so simple that I don't think our minds are even capable of understanding our brains. Yeah. As a teacher, what do I think about ChatGPT? If you are running a programming class, but you don't actually want to teach programming, then ChatGPT is cool. Otherwise, ChatGPT really worries me because I don't know if you can tell, chat, but I kind of like communicating. It's kind of a thing that I like doing. I like writing a script, and I like changing my script in a way that sounds engaging and accessible and interesting and human, importantly, right? I want my words and sentences to come off in a way where it kind of hooks you onto a story. I love storytelling. That's why I'm a dungeon master for a group of my friends. The art of storytelling, if you rely on ChatGPT, will be lost because you're just outsourcing your mind muscles to a machine, and that's no good. And so if you lose the art of storytelling, you also lose the ability to convince people that you're smart. If you always use ChatGPT to do your resumes, to do your interviews, when you talk to another human, they're going to be able to quickly understand. So here's the thing, here's my plug, right? You can be very smart, and you can be the smartest kid in class, get all A's, but when you reach a high level and you have to work with people, they're going to judge you based on how you communicate. If you communicate in a very efficient way, if you communicate in a way that is easy for them to understand, then they will think that you are easier to work with. That is a professional bonus. But if you never practice the art of communication, then you are going to run into issues where you are saying something and then you get misunderstandings on the other end, or the other person has to work too hard to understand what you're saying, and it sucks. So don't use ChatGPT. Practice communication because it makes you seem smarter. That is my ChatGPT rant. Is there anything in my studies that I found difficult to grasp? Developmental biology was very hard. As I mentioned earlier in the stream, you have to keep so much information in your head at the same time in order to see the whole picture, and my brain was not built to keep that many things running at the same time. But it is very, 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 very beautiful. I also did very badly in organic chemistry too. I got a C. That was my lowest grade in undergrad. So I found organic chemistry to be very difficult. A lot of STEM has a common way of thinking, so looking into other areas is really useful. Yeah. I think that I like biology a lot because it is very accessible for problem solving, because the living organisms are systems that you can troubleshoot essentially, or study in ways, and you don't have to have an understanding of circuits or capacitors or zeros or ones. You don't have to know any of that to start understanding biology. That's why I think it's the most accessible science to start to think scientifically, which is maybe a controversial thing to say, because I know, Saikime, you are a psychologist. I think biology is more accessible than psychology with regards to teaching scientific thinking, and I'm curious as to what you have to say about that, because you might have a different perspective than I do. Yeah. Molecular genetics, for me, things go from 0 to incomprehensible really quickly. Yeah, true. Like, you start off with Punnett squares, and that's all well and good, and then you start getting into linkage, and then you start panicking. Segregation, linkage, meiosis, recombination, math, genetics. I'm not looking forward to talking about genetics at a molecular level, because it's something that I have to study a lot on my own before I'm comfortable with talking about it. Do I have a Patreon? I'm willing to join. I actually don't have a Patreon, but I do have YouTube membership, because I want to keep all of my income streams solely on YouTube, just for the ease of my channel. And YouTube, while YouTube does take a cut, it's not like a big cut, and it's pretty convenient. So I actually do like being on YouTube for support. If you want to support, you don't have to, of course. I have a full-time job outside of this. I'd argue that there's actually some pretty big differences between science and engineering mindsets, complementary skills almost. Yeah, I had a collab with my office mate who's an engineer, where we talked at length about the different mindsets between science and engineering. I also think in many ways biology, and especially the more medical sides of it, is more engineering than science. It depends on the application, I think. I absolutely agree that biology is more accessible than psych. That's really, really interesting to me, and I'd love to hear a little more about why you say that, because I think for psychology, there's less context, maybe, because before you get into the fun parts of biology, and like for the children in the chat who have not taken a biology class, who are watching the stream and are enraptured by the content that I make, that is likely not going to be your experience when you take your first biology class. Your first biology class is going to involve a lot of memorization, and it's maybe not going to be that fun. But the thing is that you have to eat your vegetables, right? You have to do the work to learn all the context before you start getting to the fun part. I'm not going to reveal what school do I teach at, because I don't want to be doxxed. As you go up the abstraction hierarchy of science, things get harder because data is fuzzier and relationships go from more causal to more correlative. That is true. Physics is easy because there's like 5 rules and they always apply. I found it very difficult to learn physics. Psychology, especially my area, is highly context dependent. Principles that seem solid will change based on culture and time period. It's wild. Oh, that's right. So I actually had this conversation with Sidere, where psychology, while there's less macro context, I think, because before you start getting to the fun things about biology, you have to learn everything about the cell, and I mean everything. Psychology, I think, is difficult because of how much of a headache, how messy psychology is. It's so messy. And anybody who's ever tried to do psychology research can probably advocate for the messiness of humans. To design a clean psychology study, I think, is really, really challenging. And I've tried a couple times, because I've said this on stream before, but one of my potential career paths when I was a kid was I wanted to be a psychologist at some point, because I was fascinated by the work of, I forget the Cornell professor, Saikime, you might know him, the one who did the stuff about meal size, like portion size, the one that was saying, oh, you know, if you have a bowl of soup and you connect it to a tube that feeds it soup, and you are eating at this infinite bowl of soup, and you don't know that this bowl is infinitely pumping soup, you'll overeat a bowl of soup and you won't even realize. So this is a scientist that was incredibly influential on the psychology of the regulation of diet, and I really, really wanted to work with that professor. And so, as an not as an undergraduate, as a high schooler, that's where I wanted to end up. But I got rejected from Cornell, so that part of my life got shut out immediately. Wansink, the guy who had to retract a lot of papers recently. Yeah, there was some controversy with this guy. Not the most honest of scientists, it turns out. But that doesn't change this. I mean, good thing that I got rejected from Cornell, because I don't want my career to have been tarnished by working with someone that was less than honest in the sciences. Yeah. Yeah, physics was really hard. And to rant a little bit about why I found physics was hard is, man, understanding what counts and when you should apply. Here's the thing, free body diagrams. Free body diagrams and knowing when your system should be closed and open, that to me was the hardest thing, and understanding how to go from learning a formula to then being able to apply it. This is actually not a bad time to talk about Bloom's taxonomy. So Chad, do you want to hear a little bit about teaching and learning meta? Maybe I'll do a teaser for the full stream later. We could keep chatting, or I can go into a long digression about teaching and learning, and I'll poll. Teaching and learning meta. So I was trained, which is kind of unusual for academia. Most professors don't have to learn how to teach. In fact, I think no professors have to learn how to teach. They're not required by the university to take a teaching class to learn how to teach. Whereas, for education levels below university, you have to be teaching certified. So in graduate school, I made sure to get as much formal training I could to understand. It feels rude to call it the psychology of teaching, because it's not really the psychology of teaching, but kind of the science of current methods of teaching, I think, would be interesting to get into. Is Ivy League overweighted? I don't really know what you mean by that. Overrated? All universities are as good as you make your experience be, right? You don't just go to an Ivy League university and expect success right after graduating, but it does help. There are a lot of people out there that will tell you, oh, Ivy Leagues are overrated. You don't need an Ivy League education to be successful. That is true. What I will say is if you have an Ivy League degree, it is easier. That's something that you have to acknowledge. It is easier when you have an Ivy League degree. Of course, it's harder to get in, but it is easier when you have that fancy name after your degree. Of course, you can make up the gap, but if you don't go to an Ivy League, and I didn't go to an Ivy League, you can make up the gap by having a very nice resume while you're an undergrad. Work for all the labs that you can work for, apply for all the internships that you can work for, do your own independent projects if nobody takes you in. Like in college, you have to grind, right? College, if you want a job that is technical in nature, you don't have to go to college, right? You can get certificates, but if you want something in academia, you have to be grinding during your undergrad years. That's just unfortunately the reality. Okay, so people are saying that they do want a little bit of secret info in the teaching and learning meta. So let me go ahead and grab a certain image to share with you all. And I'll use physics as an example of ways that this system can be difficult to implement. So when I was learning to teach in graduate school, I'd been teaching for a long time. I've been teaching, I did tutoring when I was in middle school. I dropped teaching in high school for a little bit, but then in undergraduate, I taught almost every year I was an undergraduate, either as a TA or as a supplemental type of instructor. Because again, I like sharing things that are cool. So it wasn't really a question. I was going to figure out a way to share things that I find cool, and I was going to get paid for it. Which is also an interesting discussion that maybe I'll return back to after I talk about Bloom's taxonomy. So chat, have you heard about Bloom's taxonomy? Let me put up a poll. Bloom's taxonomy. This is really interesting. This is how we learn academic information at the very least. I don't really think this applies to skills, but it applies to things like biology very, very well. So have we heard about Bloom's taxonomy? I'm seeing a lot of yeses. Is there a difference between this and taxonomy? Yes, Bloom's taxonomy is not the same as what taxonomy means in biology, right? Taxonomy in biology is not Bloom's taxonomy. Bloom's taxonomy is specifically a teaching tool. So you might have to revise your answer if you have seen that. So let me go ahead and put up this image. I have to find it in my drive. Bloom's taxonomy. In the future, you will have more subscribers. There'll be hundreds of messages per second. Imagine trying to read chat then. No, it'd be crazy. The thing is, I don't know what my maximum potential for a live stream audience is. I certainly want to ride out and, you know, get as big as I am comfortable with. But I do sort of wonder what the upper limit of this experiment in streaming content is. It's kind of a little unusual, I think, to be streaming like this. Okay. So, Bloom's taxonomy is a teaching tool. It is this teaching tool. I pulled this graphic from Wikipedia, and it is roughly the structure by which we learn. So, at the bottom here, you have things like knowledge and comprehension, and that upgrades to application, analysis, synthesis, and evaluation, right? So, knowledge is just knowing things. And so when you first take your first biology class, you are going to be getting a lot of knowledge, with very little anything else, right? It is just the facts. Comprehension is a little more than that. It's knowing the facts and then knowing what those facts mean. And so as you climb this ladder, you start to get closer to the fun stuff. The fun stuff is synthesis and evaluation, right? So I don't want to get too deep into this because I'm going to have a whole stream just on teaching and learning meta when the school semester starts. But the thing about physics class is that lectures would often give us the knowledge. It would tell us the formulas. And it was really hard to move beyond knowledge. Like, I would know the formulas, but how to apply them is a skill. And that was really difficult to acquire. So my advice to people is go to office hours. People are more than happy to tell you what the answers are to help you make those connections. And so, if you want a kind of peek behind the scenes, and this is really helpful, like if you don't make it to that learning and teaching meta and you only make it to this one, I would advise you to use this idea as a troubleshooting tool for when you yourself are struggling with understanding a concept, right? Like, you can use this tool as a teacher to diagnose why a student doesn't understand something. So there was a student that I was very surprised didn't understand something very basic about biology. I was actually shocked because, you know, this is a university class and there was something very, very basic that they did not understand about how cells worked. And so to keep my cool, I thought about Bloom's taxonomy and I understood, okay, I need to diagnose at what point did the student's previous education not stick. And so the way that you can use this tool as a teacher or just as yourself as a student is to go down the ladder to check whether or not you have the required tools. So I think they were confused about what it meant for a gene to be upregulated, which to me is by the time you get to my class, you should know that you should know what upregulation of genes means, right? But I thought to myself, okay, so upregulation of genes is a process, and I need the student to understand this process at the application level because we're using genetic manipulation for our system. And so I needed to go back to understand first if the student understood what it meant for a gene to be upregulated, just did they understand the concept of genetic regulation? And then failing that, going all the way to knowledge, just trying to check whether or not they understood how like proteins were made, like how the central dogma works, right? So you can kind of see that there's this hierarchy of understanding. And the reason why I struggle to call this psychology is because I don't know that this is like scientifically proven, but what I do know, and something that I'll cover in the teaching and learning meta stream, is that teachers that apply this tool see better results in their student. Like their exam scores are higher. So this works, even if it might not be real, it works, and that's good enough. Yeah, it's a neat diagnosis tool for education. Yeah, it's true. Like, you can be stumped. I've been teaching so long that it's hard for me to take myself back to a time where I was legitimately stumped by a student, because you just get really quick at using Bloom's taxonomy to diagnose why students have trouble understanding. But I remember at the beginning, it would be a lot of kerfuffling for better or for worse. I'm a college freshman first year this year. That's very exciting. College is awesome, but it's also very difficult, and a bit of unprompted advice would be to, and this is a problem that I've been seeing a lot in my classes, is that if you are struggling, do not wait to get help. Do not wait to get help. I say this every stream. The second you are feeling like you are in hot water, a lot of universities have paid for therapy. A lot of universities have counselors that will help you set up studying strategies. A lot of universities will help you do what makes the most financial sense for you, because despite universities taking your money, especially in the US, there are a lot of good people behind the scenes that don't want you to be wasting your money on semesters where you will not be operating at your maximum capacity, right? So do not be hesitant when your grades are not where you want them to be. Don't try to do it on your own for that long. That's my number one thing that I see behind the scenes. I wish I could just like grab some of these students by their shoulders and just shake them and being like, get some help. But as an adult, right? Like if you're in high school, you don't have as much autonomy. If you struggle and you're in the red, your teacher will set up a meeting for you. In college, you don't have that. It's up to you to be your own advocate at that point. Is it true that academia and publishing papers is insanely difficult? It is. I've actually never published because none of my stuff actually worked. Ha 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 ha. And then I became a teacher. But I became a teacher out of pure joy in terms of like, if I had published and became a scientist, I don't think I would be this happy, because I can afford a computer off of a YouTube career that you have all helped to build. Again, very thankful for all of you watching and for the members helping fund this beautiful computer that I can now stream with and without any lag or delay. So I think I was always meant to be teaching. It's truly such an amazing career to have, especially because in the US, teachers who teach high school don't get paid very well. But if you teach at university in this specific type of position, you can actually survive, which is really, really important. Which is also why, like, my principle is to reinvest the money that you give me into making content. But I'm not going to go into debt over this channel, is what I'll say. To be fair, a lot of education frameworks are psychologically informed, even if they're not tested using empirical psych methods. Yeah, and the thing is I was such like a little weenie back when I was learning this. I was like, actually, is this actually like real psychology? When I first learned about this, I met it with a lot of skepticism. But after I started implementing this tool in my own practice, I realized that it doesn't matter if it's real. What matters is that it works. Never wait for help. Foundational maximum will always pay dividends your whole life. Don't hesitate to get help ever. Yes, I will probably say it again in the teaching and learning meta stream. I am very skeptical of therapists and such and have never found them to be helpful. But academic assistant, that kind of thing, worth its weight in gold. So I'm sure you've heard of this before, but it depends on what therapist you get, whether or not you're a good match. I myself have had therapy, especially in grad school. I had more therapy than I've ever had in my whole life because grad school is difficult. We have a couple of people currently in grad school in the chat right now, and they can also tell you it is very difficult. Managing your anxiety in the long term can be a huge burden on your mental health, and seeking help is very good. And so what I'll say is that having spoken very deeply with therapists at a meta contextual level, not necessarily as like their, I guess you can call me a patient or whatever, but as like colleagues just interested in their practice of therapy, what I will say, what I have heard and what I understand is that therapy is not very useful because it is more Socratic than something like a life coach where it's more prescriptive, because a therapist cannot tell you what to do. A therapist is trying to help you figure it out for yourself, and so it can kind of feel not very useful, but for me that was actually the approach that I preferred. Actually, that's not true. When I first started therapy, I wanted to just be told what to do. I explicitly told my therapist that I wanted behavioral therapy, like cognitive behavioral therapy, that I was feeling a certain way. I wanted to stop feeling it. So I wanted coping strategies to deal with the emotions. And thankfully, my therapist did not listen. My therapist helped me. I'll maybe say a little bit of it. I was very insecure about my own intelligence, which might be surprising as I run a whole channel for STEM content. But I actually have a very thin, fragile ego because of the way that I grew up. I grew up in a very intense academic environment where even though I was performing very well, I was never near the top. It always felt like I was a second-class citizen in a group of very high achievers. And so even though I put in so much work and I felt like I deserved to feel smart, I was never allowed to feel smart because I was surrounded by these geniuses literally all the time. And so I noticed I was like reacting very negatively to having any aspect of my intelligence challenged. I would like, it would be like triggering, like legitimately I would feel overwhelming emotion when it comes to not feeling smart. And therapy, I just want to be like, what can I do when I start feeling these feelings? What can I do to like cope with that? And which is a very like masculine coded way of thinking about emotion, right? Like I didn't want to deal with the problem underneath. I just wanted to deal with the emotion. So my therapist helped me to come to terms and feel a lot more secure in how smart I am. Which again, it's not that I feel like I'm smart. What I think I have come to terms with is that there is a level of intelligence that I cannot achieve because it's just not like born that way. I don't want to talk about IQ in this stream, but like there's a maximum. My GPU, my internal brain GPU runs, it has certain specs, and those specs are biologically encoded. But what I can do is work harder. And what I like about myself is that as long as the work is nice, as long as the work is interesting, as long as I care about the work, I am capable of putting a ton of work into things. So I make up for my perceived deficit in intelligence with just pure unadulterated raw passion. Just doing the work because I love doing the work. And to me, that made me feel a lot better because, I don't have to be genius level smart to make an impact on the world, because there's a part of me that loves doing this because I love doing it. I hope that makes sense, because I've seen this attitude a lot amongst men on the internet that like, therapy is not very, and I'm not calling you out specifically. I'm just echoing other perspectives that I've heard about the downsides of therapy, is that life coaching, if I had gotten life coaching, I would have been coping. I would not have been thriving, and maybe I'll say. Get some help. I just turned to Hollow ID for a sec. True. Get some help. I love that. I love that. I make less than the local high school teachers. That's great. Yeah. Unfortunately, I don't know that psychology has similar positions, psycheme, in terms of well-paying academic teaching positions, because every field is a little different. And I feel very blessed to be in a field that the government cares highly about, given the last COVID outbreak, right? Biology will be well-funded. My belief is for a very long time, because you know, we live in the age where COVID did happen. And so now that that reality is set in stone, I don't think people are going to be very comfortable with peeling back biology funding. How often do your students ask questions that you are unable to answer? Pretty often, I'd say, which I love. I love the feeling that I can impart on a student where you have pushed me to the point where I no longer know the answer, and that makes them feel smart. Except for the kids that get like a kick out of that. Those kids are very annoying. When they get glee, when they get glib about knowing something that I don't, those students are the worst. Actual serious advice: how does one find a life coach who is effective and not just BSing? I would not recommend life coaching as a solution, is what I'll say. If I did CBT, confirmed. That is true to some. You can say technically, more juice. That is correct. I did do CBT. Sorry, children. Imposter syndrome sucks. Yeah, no, it's definitely true. I don't think that what I was experiencing is imposter syndrome. I think what I experienced was the true hard reality. And I'll actually I've talked about imposter syndrome before, so I apologize to my viewers who have heard me rant about it, but I think it's a really interesting topic. But I want to catch up on chat before I start ranting about imposter syndrome. The most successful friend I have is one of my least smart friends. He's still smart, but we're all smarter. He works super hard all the time, but he's achieved much more than we all have. Yeah. I mean, smartness is not the only axis by which you can exist as a human being, and smartness itself is pretty poorly defined, so it's not a really meaningful metric by which you measure yourself with. I don't think therapy is bad intrinsically. I just think it's not the right thing for me personally. I don't know about enough alternatives to therapy to suggest anything else. I'm also not a qualified mental health care professional, so I'm not going to step into that realm. I'm just speaking from my own personal experiences that therapy is hard. It can feel like you're not making any progress, and it can feel like the insights are not valuable. But if you find the right therapists, I think it helps deal with the roots of certain problems in a way where I feel like life coaching does not rise to that same level of ability. Getting stumped is also a great way to create new connections in your knowledge base. That's where really amazing stuff lives. Don't mind the Socratic method. Yeah, Socratic method is also a lot of the teaching training that I was given, learning how to ask good questions. And the thing is if you're a student in my class and I ask you a question, it's terrifying. But if you're chat and there are no stakes, it's actually really fun, right? I think chat would agree. I've had a lot of fun. But I remember being a student, and it was just an awful experience being on the other end, because I felt like, again, the thing about having insecurities is that I felt like if I had gone wrong, that it would reflect negatively on my intelligence. When in reality, it's like, oh, you got it wrong. You just didn't know it. You just didn't come across that information, which is not an issue. Yeah, so let me talk a little bit about imposter syndrome. So I was trained by someone who had gotten all of their degrees at Ivy Leagues, and they had imposter syndrome. They had told a heart-wrenching story about finishing their PhD defense at an Ivy League college and overhearing at the top of this spiral staircase how great of a job they did, and then crying because there was something in their brain that would not allow them to accept the fact that they had done a good job, because they had lived so much of their life with imposter syndrome. Now, this is a person that has been educated at Ivy League institutions almost at every level of their education, like incredibly intelligent, incredibly polished thinking. I actually owe a lot of my own teaching theory. Like, this channel would not exist without my advisor, because my advisor imparted so much clarity in their method of thinking. All of these journal clubs is because they ran journal clubs that were very disciplined and very tight with regards to critical scientific thinking. And I loved those journal clubs so much that I wanted to do it for the general public, because why should people in academia be the only ones to work through a scientific problem like how scientists do every day, right? Like, why should we be the only ones to get to play with ideas this complex and come to conclusions that profound? Everybody should be able to have that opportunity. But the fact that they experienced imposter syndrome was crazy. Now, do I think that I had imposter syndrome with regards to my own intelligence? I think that it depends, because I feel like when I was in high school, I was working hard enough to be Ivy League material. That I put in just as many hours, if not more hours, than my more smarter friends. So I felt like I deserved Ivy League, I guess, in a way. I don't anymore. For various reasons. But again, just as a perspective in terms of my own relationship with imposter syndrome, I also wanted to be a medical doctor. And so I wanted to be a medical doctor, and I felt like I wasn't good enough, even though I was working as hard as I could. And I felt not intelligent because my grades were not reflecting how hard I was working. And that made me sore. It made me kind of a bummer pre-med, right? But actually, I've had friends point out that I would not be a good doctor at that age, which I took very negatively. Like, what the heck? You don't believe in me? Like, you're not being a supportive friend, et cetera, et cetera. But in reality, I would be a terrible doctor. I was experiencing imposter syndrome because I was an imposter. I could not foresee with enough insight what it meant to be a real doctor. And in retrospect, doctors don't have enough free time, man. Doctors work so hard. They work all the time. Their jobs are not that extrinsically rewarding. Like, sure, you get money, but what is money without time? Go see my collab with Psychen about all the downsides to living as a real doctor. So it's kind of interesting because I on one hand, you could say that I am smart. I would say that that is not true, because I would say what's more accurate is that I'm passionate about what I study, and that enables me to work much harder. If I were smart, I would be able to do the same amount of work in a fraction of the time, right? I would not have to watch all these videos, write all these notes, rehearse all these facts about biology, take all these practice tests. If I were really smart, I would just show up to lecture, hear it one time, and then understand everything immediately. So I don't think that I'm smart. I think what it is that I love biology so much that I'm willing to put in the hours to get good at it. And so I have come to terms with that, so I no longer feel like an imposter. But it was really rough all those years, feeling simultaneously like I deserved to be good enough, but then not being good enough. It was really tough to keep those different perspectives in my head at the same time. Imposter syndrome story resonates. I went to the worst college in Canada. Most of my colleagues are Stanford, Cal, MIT grads. Took a long time to get over being intimidated. Yeah, no, I definitely feel that. I felt like in the crop of graduate students at my graduate school, I felt like I was the dumbest one, because everybody's resume by the time they get to grad school is like stupidly prolific, and the only thing that I had going for me was I did a lot of teaching, and I was able to get a couple of grants on my own, which is not a easy feat, but it's not as impressive as like, oh, I'm an undergrad that published. Like, that's obviously much more impressive than getting a small grant. Maybe it's not that much more impressive. Maybe that's more luck of the draw. I don't know. It's tough to compare apples to oranges, but I did feel the least qualified out of my cohort. But there's always that one guy who says, but are you even good enough for imposter syndrome? That's kind of how I feel with like where I was rating my own intelligence, honestly. That's kind of funny. Sounds like a great advisor. It's true. She cried when I gave up finishing my PhD, so it's kind of awkward to reach out back to her, but maybe I will, just to thank her for setting my brain up very well. Like, even if I didn't become a researcher, even if I will never contribute to science by pushing it forward, I can thank her for allowing me to propagate the clarity in her thinking to my students and my chat. Maybe that'll make up for the disappointment she surely felt when I decided I didn't want to finish. Why don't I publish papers? Too hard. Science is very, very difficult. And also COVID happened during grad school, delayed research by 2 years, and I didn't want to stay another 2 years. Do you play video games? And if so, what are your favorites? I love playing video games. My favorite right now is Slay the Spire. I love playing Slay the Spire. Celeste is a good one. Warcraft 3, The Frozen Throne. Brood War, StarCraft. You can kind of tell my age by all these games that I'm listing. I actually think I got a better education because bad schools had low standards, so there was no pressure, and I knew I'd get good grades. Yeah, I kind of feel that way too, where like, I don't know that I would send my hypothetical child to the same schooling that I got, because I was such in a pressure cooker environment, and I'm a very go my own pace kind of person. The harder you have to work to learn something, the better you are at teaching it. Yeah, that's why I think like you know, these genius kids that came out, when they try to explain something to you, it's not actually very good because they haven't had to teach it to themselves all that much. The more meta you get into it, smart is such an inadequate descriptor. Yeah, exactly. That's why I refer to smart as being imprecise, right? Like smart can mean so many things, right? If I went to a mechanic and I had something wrong with my car, and they start telling me things about spokes, I have no idea what they're going to be talking about. And mechanically, I don't think I would be very good at understanding how a car works either. Everybody has things that they're good at and things that they're not good at. And I think that is a better metric or a better framework to think about quote unquote intelligence. Taylor, thanks for us too. That's very sweet. I mean, yeah, I mean, again, I've been sort of avoiding. It's been a couple years and I haven't reached out at all because I felt so bad. Yeah, I was crushed watching her tear up when I told her that I've had enough of this. But I think I am happier than ever. If I were a working scientist, I would have like no time. I would have no time and I would have no anxiety-free time. Because if you are a researcher, you live in a constant state of anxiety because you never know where your money is going to come from and if you're going to be coming into work the following day. Sure, there are grants, but grants run out and public interest in your idea might run out as well. So while biology as a whole might be well-funded, certain fields in biology, it's kind of touch and go depending on what you study and whether or not you've had a publishing record. And you also don't get paid very well if you study basic science and you do basic science. Speaking of TF2, now got rid of bots finally. Members gaming when 5. That's true. I did say summer vacation would be the time to game, and I do have a computer that could actually handle that. So we'll see. We'll see. I think definitely I want to do some kind of members gaming thing over the summer before classes start and I really have to go back to work. The pain of grant writing and the semi-randomness of it. I actually really liked writing grants. I got rejected the overwhelming number of times I attempted, but like there's something very satisfying about writing a piece of literature, I guess you could call it, that describes how awesome your project is. And if you're really good at it, you start to buy your own, what is it? What is the term? You start to huff your own fumes or something. Because sometimes I'll like think about a project and be like, oh, my project's not that great. And by the time I finish writing that grant, I'll be like, damn, I'm awesome. Actually, I'm a genius. It's really funny. Asking for help is uncomfortable for me. I get embarrassed and a bit stressed. My introvert self making me look like I have social anxiety. There's no better way to get over that than to just do it anyway. A lot of being an adult is just acknowledging that you have anxiety, and then the process of maturing into an adult is taking care of yourself in despite of that. So, you know, I do relate to that a lot as someone who, you know, I watched Inside Out 2 with Toxic the other day. And it's not really a spoiler because I believe it's in the trailer, but Riley in that movie develops new emotions, and one of those emotions is anxiety. And I told her, I think my principal controller is anxiety, but I think my anxiety killed my ennui because I'm never bored. When you are passionate about so many things, you never find a moment where you truly feel boredom. So believe me, I am governed by anxiety, but the process of being an adult is practicing the things that you need to do to overcome that anxiety anyway. The peer review process too. I know it's very necessary and important, but it is tough. Yes, that is true. And one only needs to step in a journal club to see how mean scientists can be to one another. And they get very snippy about how certain things are done. Oh my god, Reviewer 2, my greatest ally and worst nemesis all in ruin. True, Reviewer 2 always has a chip on their shoulder. I'm pretty sure I'm going into industry instead of academia because of the reasons you mentioned, even though industry is its own can of worms. Yeah, industry is also tough. It's not easy either way, but on the plus side, you do get money, and money is cool. I will never shame anybody for picking money over following quote-unquote their passion, because I had this interesting discussion with a couple of friends the other day. Chat, do you differentiate between what a hobby is and what a passion is? kind of curious as to whether or not these two ideas occupy different things in your brain. And then Crafter says no. Yeah, it's kind of interesting because I was describing. The thing I was chatting about was, I think there's something not necessarily broken inside of me, but maybe some neuroses I have about hobbies where I don't think that I could do a hobby without the potential of monetary return. Not necessarily to be able to make a job out of it, right? But I don't think that I would do YouTube unless I could get ad revenue, which is kind of messed up. And I think that's messed up. But I know myself enough to know that without the incentive of ad revenue, there's not a lot of chance that this channel exists. And so I wanted to think about and talk about why I thought that way. And it's like, is there something wrong with me where like I can't have a hobby unless it's profitable? And don't get me wrong, I run this channel at a loss. Like, you have all helped tremendously to build this computer, but I still took a hit in my savings to build this computer, which I don't mind because, you know, it's making the pre-on video so much easier to edit, for crying out loud. It is so good. But what my friends were saying is that, oh, you're talking about a passion, not necessarily a hobby. Yeah, Clockwork, that's what my friends were saying, was like, a hobby is just no thoughts, it's just nice to do. And I'm like, I guess I count gaming as a hobby, but I might be like gatekeeping the idea of a hobby. Because when I think hobbyists, I think, oh, you know, if you're a computer hobbyist, then like you build your own PCs and you get very geeky about the hardware that you use. Whereas gaming, am I playing the latest games? No, I play Slay the Spire for hundreds of hours, and I play Civ 6 for hundreds of thousands of hours, and I play the same game over and over again because it's comforting, and which is why like I struggle to think of myself as a game hobbyist, because I'm not buying the latest GPU to play the latest game to get the best specs, because that's what it feels like hobbies are. Like you take it on in that kind of way. But as they pointed out, it doesn't have to be that way. You don't have to treat hobby that intensely. And I'm like, okay, fair enough. So it's more accurate to call my YouTube channel and this science communication more as a passion. And to be monetarily rewarded for that passion is nice. But I think there is something very interesting where, like, even though I love doing this, if YouTube didn't offer ad revenue, I don't know what I would have done. I would have maybe tried to monetize this off of like Apple Podcasts or something, which I did before. Fun fact, before the history of this channel, I did start a podcast with the hopes of monetization at some point, but I didn't like doing the podcast as much as I like doing this YouTube channel. Hobbies are something that you actually do a lot, while passion is something you like a lot. I think that's fair. That's true. Because I don't game enough for me to describe myself as someone who is passionate about gaming. I just like playing Slay the Spire a lot. That makes sense. Passion, source of stress. That is true to some degree. I'm having a good time with the pre-on video now, but there are videos that I made on this channel that I'm not very happy with, and that's made me kind of sad. Also, Clockwork definitely knows the pain of weird comments in the comment sections. That has been a minor source of stress. And I'm wondering if chat knows what I'm talking about, specifically for my channel, not in general, but specifically in my channel. Yeah, because while my streaming audience is very lovely and it's a lot of fun chatting with all of you, I think my video audience is actually quite different audience than my streaming audience. And I think that my streaming audience has overlap with video, but I don't think video has that much overlap with stream. Weird comments. Yeah. Only I get to see all of them because YouTube Studio allows you to see all the comments on every video. But there are a couple of weirdos, a couple of anti-science weirdos that pop up on my channel. I've struggled with the idea of like doing a stream where I like compile all of the conspiracy theory stuff, but you know, conspiracy theorist researcher that you may be acquainted with, Sidere of V-Theorem fame, has advised me that maybe I should just delete the comments and not show them off, which I think I agree with, because even though if I'm pretty secure in my streaming audience being relatively educated and with the times, I can't guarantee that, and I don't want to contribute to weird conspiracy theory spreading. The same weird class of comments that I complain about, I thought those folks were only in my videos. No, they're in my videos too. Although, they're more common in your videos than in my videos, I'll say that. Anti-science weirdos reminds me of a friend who also has a YouTube channel focused on evolutionary biology, and they always have a ton of creationists in the comments. Yeah. I don't have that many creationists in my chat or in my comments, and let me be very clear what the official stance of this channel is. I don't care what you believe in, what happened before the first cell was formed, or before the Big Bang. I don't care. You can believe anything you want, and I'm chill with that. So it doesn't really bother me all that much, because the types of comments that I get from creationists are people overreacting to things that are not even making fun of creationists. Like, I think I had like a joke in the rabies video about what was it? I said that there is no God here, only natural selection. Like, that's barely a joke. Like, it was a throwaway gag. And you would be surprised at the number of comments that got really upset that I had made that comment. It was more like natural selection is what drove rabies to be this terrible. And that, whether or not you believe in a higher power or not, is a law of nature, right? You can at the same time believe that a higher power had created everything and still believe that natural selection made rabies so terrible. It's not incompatible, but those comments, I think, are not even the most stressful ones. Those are the ones that I feel comfortable enough with sharing those, because I think that's just like, that's light and fun in a kind of weird way. There are more that are like, man, it's wild YouTube comments. As another conspiracy theorist researcher, I agree. Psyche, are you also a conspiracy theorist researcher? That's so interesting. I'd love to hear more. The friend I mentioned recently did a video addressing the creationists, and I loved how they said, if you want to prove me wrong, just grab a shovel and search for fossils that don't fit in the layer. Yeah, I wanted to do a stream about Darwinism. Not Darwinism. I wanted to do a stream about Charles Darwin and Charles Darwin's communication ability, because Charles Darwin had riz. Now, Charles Darwin might have married his cousin, which is gross by modern standards, but every letter that Charles Darwin has sent, most of the letters Charles Darwin had sent, have been preserved for all of eternity for people to read. And the dude had actual game. Like, he had a way with words that was so fascinating, not only in On the Origin of Species, which is the book that I wanted to talk about, but also in his private communications to his loved ones. And part of why I haven't done that yet is because I picked up a book about evolution, the history of evolution, and I found it very difficult to understand. As someone who does molecular biology and doesn't know as much about the natural part of biology, it was just something that I didn't have enough context for. Like, the fossil record, like, I accept all of these, but do I really, like, did I read all that literature? No, not really. Yeah, for sure. Psyche Mail, I'll shoot you a DM to the Discord, or we can chat. Because my pet, not pet, but like part of the subculture that I love gawking into is reading into conspiracy theories. There's something very fascinating about that whole idea. And his friendship with the captain of the quad. There's so much character there. Yeah, like the thing when Oppenheimer came out, I was like, oh, a movie about Charles Darwin would also, I think, be pretty cool. I don't think that it would raise to the epic levels of Oppenheimer. Like, I don't think it would be as big of a drama as Oppenheimer. But Darwin was an interesting guy, put it that way. And I loved his way with words. Like, not to toot my own horn, but I'm going to toot my own horn. I think that I come across pretty eloquently. I think I've had a lot of practice with formulating the words that I want to say in a way that I want to say them. And part of my inspiration is Charles Darwin. I was also inspired by Richard Dawkins and Neil deGrasse Tyson, who have become Twitter cringe lords, but Darwin, although Darwin himself was pretty problematic, but again, context, he lived like 200 years ago, so, context. But the way that he wrote Origin of Species is so beautiful, it almost reads like poetry or like some sort of spiritual text. And that can give creationists kind of fuel for the fire to say that evolution is also faith, but it's more like being able to connect a feeling of faith with, or not faith, but a feeling of spirituality with your own biological history, I think was very poignant. And I would love to talk a little bit about that in the future. Adding those letters to my reading list. Yeah, I have a whole bunch that I've like clipped the best part. And he's also as he was can I share? Can I share? Chat, will you allow me to share something very sad about Charles Darwin? And maybe this will act as a teaser for the Charles Darwin stream. I want to read his last words. Because they are very sweet. Let me just try to find that. Dang. Try to find it quickly. Chat amongst yourselves. As I try to find these last words. Duck, duck, go. Come on. That is not what I wanted. Okay. So the thing about Charles Darwin is that he was sick for a lot of his life, which is tragic. He lived a surprisingly long time, given all the various health problems that he had. But his wife, Emma, had to take care of him for long stretches of time. And I think what is poignant about that, I am really trying to find this thing. But no, you know what, maybe I'll just tease it. Maybe I'll just actually find the real thing in the... Wait, no, I can do this, because this is something that I've actually have a script for. I have a script for this, which means I have this information somewhere in my Google Drive. I just have to find it. Darwin stream. Yes, Darwin stream. This is the wrong Darwin stream. Maybe I'll find it later. That's very sad. Who would I pick to play Darwin in the movie? I don't know. That's a good question. I don't know. Maybe some actor who got known for sitcoms and then had a revival as a serious actor, I think would be crazy. New PC dying. It's not really dying unless OBS is acting funny. Incoming rain in my area. Heard lightning, but it's clear outside. Oh. Was there lightning here? It is slated to rain nearby. So I'm wondering if that really was me. Yeah, I guess I'll spitball a little bit about Darwin as I try to bring this up. Maybe I'll still try to find this piece that I have here. Is that Darwin, he was a little bit of an F-boy growing up. He was very privileged. So in terms of having a sort of sympathetic story, he's not really that sympathetic for the beginning half of his life, because his grandpa, I think his name is Erasmus Darwin, was a known naturalist. So he came from a position of academic and financial privilege. So his story is not one of like, it's not really ground up. He was an academic born into a family of doctors and academics. So it's not really engaging in that way. Okay. I think I found my document. Okay. Okay, I found the words. Oh, okay, here we go. Okay, ready for the sad time? Okay. So Charles Darwin, sick all of his life, Emma had to take care of him, and the letters that Darwin and Emma shared amongst each other are sickly sweet. Like, they are very cute, and even though they were written 200 years ago, you can still feel the riz come off of them. It's very clear that they loved each other very much, and they very rarely fought in terms of what we know from the letters, right? So towards the end of his death, it was, he was succumbing to his health issues, and on his deathbed, Emma was asleep. And so, during this time, he prompted his children who were awake with him to not disturb her. I'm guessing because he likely felt like a burden to Emma for a lot of his life, right? If you have chronic illnesses, it ends up being that you are putting work on your caretaker. In this case, the caretaker is his wife. And so his last words, as reported by his children, were, "I am not the least afraid of death." And as he was like, not hallucinating, but he's very hazy at this moment, thinks that Emma is still up with him. And he says, "Well, remember what a good wife you have been to me. Tell all my children to remember how good they have been to me." And then as he's like droning off, he says, "Don't call her. I don't want her. Said often, it's almost worthwhile to be sick to be nursed by you." And that hits pretty hard for me, I think. That this guy, he has this this coyness, almost this kind of I don't know how to describe it. Riz is not really an appropriate word, but it's a kind of humor, even as his last words, that like, even though I've been sick, to be in your care, it was worth it to be this sick, which is, I get a little teary thinking about that kind of idea, right? Because if you think from Emma's point of view, Emma, I mean, she's not as like gifted in the Riz department as Darwin, because Darwin has been an F-boy his entire life. He wrote prolifically. He wrote to everybody. And Emma, we just don't have as many of those letters. And also, women getting educated back in the day, not as much of a priority, right? But when you read the letters that they share with one another, it's so clear that Emma loves the crap out of Darwin, that like this caretaker position that she has. I can't say for sure because I'm not Emma, but it feels like Emma really, really was okay with it, that she misses Darwin when he's out, that she worries about his health when he's out, that they shared a very genuine relationship. And it's kind of crushing to acknowledge the fact that Darwin, in his last moments, simultaneously felt as if he was still being a burden, and yet wanted to convey the fact that just he was so thankful to Emma for everything that she had done for him. And that is really beautiful. And it's one of the texts in the letters that I want to cover the most, because he lived 200 years ago, and there are not that many things from 200 years ago that I think that could elicit that much emotion from me. But like, knowing the context of how much he loved her, now that you know the end of the story, I'm kind of like playing a sad movie in reverse, right? You know how this ends, and when I share with you all the letters that they shared with each other, you're gonna think, dang. Oh, I want to read one more. I want to read one more. One more. Okay, one more, chat. One more, because Darwin has the illest nerd riz. Like, you know how nerd riz is almost misogynistic in the way where like you kind of view women as an object to be obtained. And again, if you do it now in the year 2024, don't. But this is 200 years ago. It's a little more forgivable. Times were different back then. So he's got a little bit of nerd riz. And so Darwin was very committed to his job as a working naturalist, right? Studying and traveling and, you know, taking a wife, having children. That is not an easy decision for a researcher to make. So what does Darwin do? What does Darwin do? Darwin makes a pros and cons list, right? Darwin makes a pros and cons list. And I'm just going to read this actually directly from my script that I have, because I was going to make this a video, but the Prion video was more important to make. Okay, I'm going to read this word for word from my script. Here is his pro and cons list. Sanitized because, well, there are things that dude said in the 1800s that we know now are messed up. You're free to look up the letters yourself. Link in the description. There's no description here. Under not married, Darwin wrote, no children, no second life, no one to care for one in old age. What is the use of working without sympathy from near and dear friends who are near and dear friends to the old except relatives? So he's kind of contemplating the pros and cons of not marrying, right? So no children, no one to care for in old age, so you don't have caretakers when you're old. Freedom to go where one liked, choice of society and little of it, conversation of clever men at clubs, not forced to visit relatives and to bend in every trifle. To have the expense and anxiety of children, perhaps quarreling, loss of time, cannot read in the evenings, fatness and idleness, anxiety and responsibility, less money for books, if many children forced to gain one's bread, being able to make money. But then it is very bad for one's health to work too much. Perhaps my wife won't like London, then the sentence is banishment and the degradation with indolent idle fool. So the text under the not married column read like Darwin's superego, the rational thinking man coldly listing off pros and cons of living the life of a bachelor. He reflects on the potential ease of that lifestyle, not being tied down to responsibility, having extra money around, avoidance of conflict, all written in a cold and clinical manner. Time and money are particularly quantifiable parameters to consider. But once he starts thinking about getting married, under married, Darwin wrote, my thing tabbed, children, if it pleases God, constant companion, friend in old age, who will feel interested in one, object to be beloved and played with, better than a dog anyhow. Better than a dog anyhow. I want to take a break and just let that sink in with the chat. And this is a beautiful little rhetorical duplicity here. Because how does that hit your ears? Children, if it pleases God, constant companion in a friend in old age who will feel interested in one, object to be loved and played with, better than a dog anyhow. Wife, better than a dog anyhow, right? That's crazy. That's a crazy thing to say. How does that strike your ear, chat? Wife is better than a dog. Does that strike you as kind of incel-y? It's kind of an incel thing to say. I don't know. That's, to me, that's not like, it's kind of weird, right? But the thing is, it's a little bit of self-referential humor because Darwin is actually quite aware. This was writing for his own eyes, more juice. This was in the margins of his research notebook. That almost sounds the opposite of it. So I guess you have a point there. To me, this struck me as kind of misogynistic. To even compare your wife to a dog is kind of weird. But then upon thinking about it and reading more of Darwin's work, it's clear that he kind of understands that he's not, that he's kind of awkward. Like he comes across as the Rizzler in his writing, but in his interactions with people, he was actually kind of socially awkward. And he had like resting mug face, I guess you could say. He didn't like his photo being taken, which is really funny as well. So even though he's made a lot of friends, he's not asocial, but he is a bit of a nerd and he kind of knows it. And this bit about him comparing a wife to a dog, he's actually imagining a future where he is a crazy cat person, which I think is kind of fun. So, home and someone to take care of the house. Okay, it's the 1800s. Charms of music and female chit-chat. Ooh, female chit-chat. These things are good for one's health, but terrible loss of time. My God, it is intolerable to think of spending one's whole life like a neuter bee, working and nothing after all. No, no, won't do. Imagine living all of one's day solitarily in smoky, dirty London house. Only picture to yourself a nice, soft wife on a sofa with good fire and books and music, perhaps. Compare this vision with the dingy reality of Saint Marbleau. Mary, 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 Q-E-D. That's his notes to himself. In contrast with the Not Mary column, Darwin scribbles under Mary read like an internal monologue. It starts similarly to the Not Mary, quiet musings on the benefits of companionship, a potential self-dig at his own potential future as a lonely, crazy dog man. From there, his writing takes on a soft warmth, charms of music and female chit-chat. Then a defensive shift from feeling to thinking. He puts it into terms that a rational thinker might understand. These things are good for one's health. It's not that these things are nice, it's that it's good for my health and I'm a sick guy. This shift brings him back to a familiar territory. It's kind of like it reads like a psychological defense. Like, oh, I'm feeling too much. So let's think about this academically. I could be reading too much into it, but I don't really think I am. It seems very like Darwin. This shift brings him back to familiar territory. He's still worried about the results and terrible loss of time. But this tension that builds between his thinking and feeling brain builds, and we see him spiral in despair at feeling the emotional reality of his dark and lonely bachelor future. My God, it is intolerable to think of spending one's whole life like a neuter bee. Working, and nothing after all. No, no, won't do. Imagine living all one's day solitarily in smoky, dirty London house. And then he imagines an alternate future. Only to picture yourself with a nice, soft wife on a sofa with good fire and books and music, perhaps. Compare this vision with the dingy reality of Marlborough Street. Darwin hammers in his resolve in the nerdiest way possible. Like a drum, he beats Mary, 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 Q-E-D. And that's wonderful. Get you a man who calls you a nice, soft wife. That's so cute. There's no better way to describe that as very agonizingly moe, right? That is the most moe freaking thing that I've ever read. Darwin writing this joke to himself only for some YouTuber to analyze it on stream. I know. He's not alive to defend himself or exert any level of privilege over these texts, so I will take full advantage of it. But I love the fact that he imagines a soft wife. Like, that's so funny. It's so funny. I love that so much. Never thought of Darwin as moe, but here I am nodding along. Yeah, Oppenheimer is rough. That was a rough movie to watch. That's a very edgy set of circumstances, but Darwin could be a romantic comedy. A romantic comedy tragedy. Of course, we do know how it ends. Darwin Chan. Yeah. I love The Origin of Species so much that this episode script was like a prelude, motorcycle going by, a prelude to a whole episode on just The Origin of Species. But the thing is that as with my tendency to overestimate my own intellectual ability, learning about the history of evolution has actually been too challenging. I've read Origin of Species from front to back for my rhetoric of science class in undergrad, because I loved how Darwin communicated and I wanted the chance to study Darwin formally. But actually, like, learning how the fossil record and the history of the fossil record and the history of creationism and the history of the idea of evolution, that was all too much work for me, so I didn't feel like I wanted to do that. But maybe one day I'll just do nice quotes from The Origin of Species because Origin of Species is cool. It's a very well-constructed book. As a rhetorical tool for communicating the idea of evolution. You have to imagine that in Darwin's time in the 1800s, the predominant form of how people thought people came to be was creationism, given the state of England at the time. And the fact that Darwin released The Origin of Species and it didn't cause a societal meltdown is actually really telling at how good The Origin of Species is at making sure that creationists don't get mad. My final project for that class was actually analyzing the ways in which Darwin uses, what is the term? It's been a while since I took rhetoric. Polysemy. So more than one meaning, same text. The way that he uses this rhetorical duplicity to appeal to both people who want to believe in creationism and the people who don't, which makes him just a master of writing. It's beautiful. Were creationists even around back then? They definitely were. Definitely were. Ever read Jurassic Park? I have not. Yeah, I mean, Darwin did get pushback from the church, but not that much. There were a lot of prominent Catholic scientists that agreed with him because, again, creationism is actually not incompatible with evolution, depending on where you draw that line, right? As long as you say, oh, and then before any of that happened, God did this, and then everything is kosher. So the fact that Darwin had worded specific sections in The Origin of Species to not sound too atheist is really masterful, because if he just straight out said, oh, like, this means God doesn't exist, society would not have been that receptive to that idea at the time. Slightly related, but did these spontaneous generation people ever talk to animal breeders? Like abiogenesis? I'm not actually sure. Again, the history of evolution includes the history of abiogenesis, and I actually don't know too much about the history of that field either. The origin of species is not the same as the origin of life. Yeah, and that's kind of, there's this vagary that allows you to mold the idea of natural selection into your own existing belief system. My favorite biology rambler went live. Dude, you're goated. Thank you so much. Thank you for stopping by. I'm unfortunately at the end of the biology. Well, I guess I'm still ranting about biology. It's just not molecular biology, it's evolution. My dad called, so I'm coming back in the middle of a talk, but I was raised in a very religious household. I believe creationism, but the entire time I never saw a conflict with evolution. Exactly. You don't have to have, it doesn't have to be in conflict. I wonder if Darwin actually had the foresight to realize the implication of his theory in religion at the time, and thus tailored his writing to avert it. Yeah, so during my final project, I actually learned that there were a lot of different editions of The Origin of Species, and the later editions made the language a little softer, a little more vague appeals to the notion that there is a creator. And I found those rhetorical changes between the editions to be really fascinating, because that's Darwin being like, okay, if I want people to believe my thing, I have to not come off as a neckbeard, an atheist neckbeard, right? Can't come off that way or people won't accept the idea, which is really good, I think. Like, even though Darwin himself, by this point, I believe he had lost most of his faith through his study, but he did not craft The Origin of Species as a way to challenge creationism. He, I believe, kind of let his bulldog do that. What's his name? Henry Huxley, I think, was more of his attack dog, defending his ideas from creationists. Yeah. Yeah, I'm brought back to a very fond time when that was my senior year, when I'd taken all those rhetoric classes, because senior year for me was great. I took a lot of English. I minored in English, actually, because I liked communicating that much, which is, again, why ChatGPT. It's tempting to use it. It sounds good, but if you never learn how to say words good, then you might never learn how to sound smart. And if you don't learn how to sound smart, interviews might get a lot harder for you. And if interviews are harder for you, it might be harder for you to land a job. So just my practical piece of advice is learn to speak good. Learn words good. All right. I have yapped for 3 hours, which was very fun, especially fun with this new computer, reducing the latency between chat and my actual stream. So thank you for attending this stream. Yeah. The next stream, I think I'll probably take one of the membership stream ideas, either talking about the virome of the brain, determining whether or not there are viruses in the brain, not viruses, whether or not there are bacteria in the brain, or I might do a JoJo themed stream next, but my top priority is trudging through this pre-on video. I am working at such a high pace that I really do need these streams to kind of decompress, because all day, just from morning to night, I have been editing this pre-on video, and I cannot wait for you all to see it. In terms of announcements, this is a pretty big month for collaboration. I will be collaborating with Clockwork, who has been chatting this entire time here, who does wonderful, amazing 3D, I guess I shouldn't call them all 3D, but amazing animations on the molecular machinery of the cell. It's the kind of content that I wish I had accessible to me as an undergraduate. His video on ATP synthase is actually so crazy. You should definitely watch that because it's insane the level of detail he goes into and how good that video is. But yeah, I will see you all in the next one. Although, wait, let me do something real quick. There was, I believe, a chatter who had stumped me by answering a question correctly, and I will actually now follow up on my promise to gift 5 membership subs. So let me do that right now as a kind of sign off. So I said that I had a very tremendously challenging question for chat, and somebody did get the answer right. So I will be gifting, which is basically just giving myself money. I will be gifting 5 memberships for randoms. So I don't pick who these go out to. But I hope that they find their way to you. Let's see who ends up with the memberships. This is very exciting. Very exciting. So let's go ahead and let's buy those memberships. Who's getting the membership? Who's getting them? Alrighty, congratulations to Casahedron, Eliaston, Spolax, Mystifox, and Sang. Welcome to the immune system. Congratulations. Yeah, awesome. Very good. All right, that's awesome. Now Toxo can finally stop roasting me for scamming people out of this opportunity. But yeah, thank you all for coming. I will see you in the future. Bye, y'all.